Ah, ya está puesto ahí. Necesitamos el OK de atrás. ¿Estamos? Okay. Good morning. Good morning, everybody. Welcome to Madrid, first, for those that have come to Madrid. And welcome to the National Museum of Natural Sciences that belongs to the CSIC, which is the National Research Council for Science. I am the director of the museum, Rafa Zardoya, which happened to also work on genomics. Well, used to work on genomics. Now that I'm director, I'm not working in genomics or anything else, but bureaucracy. Anyway, we have, well, the two organizers, Iker and Aida, have gathered a nice program for today. So I hope you will enjoy it, those that are here and those that are connected through, because this is broadcast. I'm not sure if this is going to be recorded or not. Do you know? It's going to be recorded anyway, for those that decided that in the coffee break disappear or so. Anyway, so welcome again, and I pass the floor to the organizers so they can say some logistic and words, and some welcome words.
So thank you everyone for joining today to this symposium. It's been really fun to organize it, and I hope it will be even more fun today. So there is here overview of the program. There is also one at the door. We were lucky to secure 7 great speakers from around Europe. They are experts in various techniques of how to apply genomics to study biodiversity. I invite you to check the web of the event if you want more details, but basically that's the program. Some logistics. This event is being streamed through YouTube, so it means that during the questions, maybe the public will appear. If someone needs to go to the toilet, it's in the very end of this corridor, on the noisy corridor at the very end to the left. And we will have a lunch break and coffee break. The easiest way, it will be in the garden behind the museum. The easiest way to go is through this door. There is a secret passage, and then we go downstairs. Or you can go through the way you entered using the elevator. You can just follow people from the museum. There are many here. So I wanted to also mention that this event is possible thanks to generous funds by CSIC's Genome Conexión Genoma, who is coordinated by Crisanto Gutiérrez and Josefa González. This is a centralized effort by the Spanish Research Council that aims to foster research in genomics by bringing together researchers in Spain that work on genomics, but also doing these kind of events about the use of genomics. And in this case, we wanted to do a symposium about how we can use it to understand biodiversity, and specifically working in an environment of a museum with natural history collection. So I would like to thank several. So first of all, Aida, who has been working with me very hard the last weeks, but also all the local people from the museum that are more senior, have more experience in organizing this, as well as Noelia, Andres, and Alfonso for technical support with the IT and the graphics. And without further ado, we will start with the first talk, which Aida will introduce.
So welcome, everyone. Thank you for being here. And it is my pleasure to introduce our first speaker, which is Lisa Pokorny. She's a Ramon y Cajal researcher at the Royal Botanical Garden here in Madrid. She completed her PhD at Duke University with Professor Jonathan Shaw, and performed several postdocs in Spain and London in the Kew Botanical Gardens. She has contributed to advance our understanding of plant diversification through time and space. And she works on phylogenomics and population genomics, linking microscale genetic variation with microevolutionary patterns. Today, she will talk about the application of genomics tools in bridging micro and microevolutionary processes. Thank you, Lisa.
Okay, it's working, perfect. So the general title was Phylogenomics, Systematics, and Taxonomy. The more, let's say, ad hoc title, or what I'm really going to be talking about, is genomic approaches to the study of plant evolution. And in particular, I'm going to be talking about a technique called HyPSIC that combines target capture sequencing with genome skinning. This works. Next. Where do I have to point? No. Está. Bueno, pues el botón. Okay. I don't know why. It doesn't look like it's on.
No.
Ahaha. Sorry about that. Okay, an outline of the talk. So what's the deal with plants? A little bit of genome architecture on plants. This technique I just mentioned, HyPSIC. And then what you need to implement HyPSIC, which is to design target capture kits, how you implement HyPSIC, how you analyze the data, and then a little bit about what this symposium is. So biodiversity genomics, a few examples. So what's the deal with plants? They're autotrophs, which is super cool. They make their own food. I think we appreciate that. And they also make oxygen, thank you. They alternate generations. So let's see if the point, yeah. So you have individuals, so the same organism can be both diploid and haploid, and they can be free living, the haploid and the diploid. And few creatures out there do that, except for fungi and algae that do, I mean, algae are plants. They have a long history, more than 1,400 million years of history, the kingdom, with many innovations and now transition from the sea to the land. Something wrong? That one is not muted. It is muted. Okay. Technical. So, very interesting organisms. That said, what's the deal with their genomes, the architecture of their genomes and the dynamism they present? So plants can have very large genome sizes. These, I think we published 2023, so a couple years ago. We described or presented to the community of scientists the largest fern genome. This thing is, I don't know, yay big. And has a genome of 160 gigas, 0.54. And as you can see, so fungi have tiny genomes. You can sequence the whole thing. And most animals also have what I call tiny genomes. So you may think they're big. Some arthropods have a larger genomes. And in chordates, you can have some I think they're called mad dogs or mad puppies, something like that. And fishes, lungfishes, can have really big genomes, 90 gigas. But in plants, as you can see, the average genome size, especially in ferns and conifers, it's really large. And then they can have extremely large genome sizes. This poses a problem when you want to do genomics, of course, just the size. So how do they end up with these really big genomes? Part of it has to do with the repeatome, so awful word, but it's there. That's the word. So repeated elements were first described by Barbara McClintock in corn, and she earned the Nobel Prize. We've found them in all organisms ever since, and in plants they just go crazy. I mean, they get really massive. Why? One of the things that explains these expansions of repeated elements across the genome of plants is that they undergo polyploidization. So they can duplicate their whole, they can fail to reduce the gametes during meiosis. And so when you have fertilization, you have to not reduce gametes, and you end up from a diploid, you end up with a tetraploid straight away, so instant speciation, if you want to think about it that way. So once you have a tetraploid, you have double the amount of DNA. That opens the opportunity for evolution to operate, but the genome is often not stable, so many things can go wrong with a genome that has duplicated itself and will go wrong, actually. So there are like two conflicting hypotheses saying that either you go polyploid and then you go extinct, or you go polyploid and you radiate. So many things can, and what happens is that genome needs to get stabilized. So there is a very strong selective pressure for reducing that genome. So you end up with what is called polyploid diploid, so polyploidization cycles. So if we go back, so it's more or less like this, a funnel of polyploidization cycles. So when this happens, recombination is suppressed. So recombination is a very good way of getting rid of repeated elements, but this is no longer happening in plants once they go polyploid. All these mechanisms sort of stop, because if you start recombining a genome that's not stable, you die. So all of these repeated elements, they just go like, whoa, let's go massive. But that's not all. Now you have a polyploid, or you're just diploid. And we've heard, I mean, and plants can hybridize, and they will hybridize. And then you will have lateral transfer of DNA. And once they've hybridized, that hybrid may not be stable. So one way of making stable a hybrid is to go polyploid. So this leads to what it's called apomictic complexes, which is chaos. So basically it's what you've all heard of vegetative reproduction. What's happening with vegetative reproduction is that sex cannot really happen because stuff went wrong. So they go, they go for a conservative approach. It's like, let's not do sex, let's just keep it asexual and live long and prosper. So you've seen that plant genomes are complex. They're just not, like diploid, big genome, and on. So many things are happening. And, like if you're gonna assemble a genome of one of these allopolyploid apomictic plants with gigantic genomes, you're gonna have issues. So I really love this technique. A little bit of a background. So you can subsample your genome. Rather than go for the whole thing, you can go for anonymous regions, so do a low coverage, whole genome sequencing, or you can do RADseq, GBS. The whole GBS RADseq family is massive. There are all kinds of implementations and ways of doing it. This is from a paper from 2018 summarizing all of them. And they've proven wonderful, especially at the micro-evolutionary scale, so population genomics. But again, they're limited mostly for micro-evolutionary scale studies. You can go targeted. By targeted, I mean regions that will be known or are already known. So when you do a transcriptome, you're going for coding sequences. You may not know what coding sequences you're getting, but at the end, you're going to be able to figure out what coding sequences you have, especially if there's a nearby genome that has been annotated. And then there is target capture. Target capture can be solid-based or in liquid phase. And I'm going to concentrate mostly on liquid phase or liquid solution target capture sequencing. But what I said is I implement HypeSeq, and HypeSeq actually combines genome skimming with target capture sequencing, which is very neat because you get the best of both worlds. So with a low coverage whole genome sequencing, you get a high copy number variation on your genomes. So you can get all the organelles, so whole plastomes, mitomes. And with target capture sequencing, you're going to go for known regions. So you have to have a prior knowledge in order to design target capture kits. So if we compare, for instance, Sanger sequencing with HiveSeq, with Sanger sequencing, you're short of fishing, one gene at a time. With HiveSeq, you're sort of dragging a net. Your target capture kit is that net, and you're dragging it and getting your fish school of interest and then a bunch of other things that were around, okay? And the crazy thing with HiveSeq is that you don't need, so for here you need the whole fish, it has to be intact. The fish can be chopped into bits and you will get it still, okay? So it's sort of gore, but yeah, it works. So how you design your kits? So you need a bunch of resources, of course, like everything in life. Human resources, both for the wet lab. This is a course we implemented at the Botanic Garden a couple years ago. I will eventually repeat it to learn how to do all the genomic library preparation and the target capture itself. And then there is the dry lab. This is the phylogenomics workshop at Český Krumlov. It's coming up. It will happen in January, February next year. Then you need genomic resources. As I said, there are regions, known regions that you have to have available. So the way I got into this was through a quote-unquote side project I got involved in when I was doing my PhD in the States, which is the 1,000 plant transcriptomes. So by the beginning of this project, this was a pilot paper. We had 80 transcriptomes, 20 genomes, so 100 tips on the phylogeny, and we identified more or less 800 orthologs. By the end of it, we had 1,300 transcriptomes and double the amount of genomes, and we reduced those 800 to 410 orthologs shared across Archaeplastida. So this is all the land plants plus algal lineages in the plant tree of life. This is a really good starting point if you're going to do a target capture kit. But now we have genomes. There's a bunch of initiatives out there. So following from 1KP, we got funding for this Open Green Genomes initiative. So it's just 35 genomes across the diversity of plants. Then there's this 10,000 plant genomes from China that it's coming slowly. And then there are others like ERGA, Darwin Tree of Life, that are part of the Earth Biogenome Project. Of course, some folks have done a lot more careful work of curating orthologs, which is what we need for phylogenetic reconstruction, especially at the species level. And you have a large collection of orthology databases out there. This is the one for plants, and this is the one for fungi, and then the rest are mostly prokaryotes. Sorry. Of course, you also need computational, so hardware and software. Notice the hardware were computing clusters. Software, there's a bunch of tools that I've used through time, but there are many others who mostly clustering algorithms of different types. And then money. So these are projects, oops, that went out of place. These are projects I've collaborated in through the years, starting with 1KP and a bunch of other things. So a few examples of custom kits for not so big lineages, and then what I call universal kits for much larger sorry. This is totally out of place. Oh, well, anyways. So this was the first kit I designed with Isabel San Martin and Enrique Rina at the Botanic Garden. We had this issue with 3 subspecies of species Euphorbia balsamifera, and we just couldn't do anything with tRNL, which is plastid, and ITS, which is ribosomal nuclear DNA. And so we designed a kit So basically, it has 400 orthologs, and the kit has 20,000 probes that are tiled, so they are overlapping, covering this matrix, and that's the size of the matrix. And what is crazy is we were able to see very clearly not only were there 3 subspecies, they actually were species, and they had long diverged. And the crazy thing is that the one found in coastal Morocco and in the Western Sahara and the Canaries is most closely related to the one found on either side of the Sea of Aden. And both of them are sister to the one found on the Western Sahel. So these are called tabaibas in the Canaries. They're pretty lovely. Then this was another kit. So this kit here only has single copy nuclear orthologs. This other kit was done for yams. So it's a smaller group rather than 2,000, 4,000 species, it's just 300 of them. So see, it has less single copy genes, but we have genes of agronomic interest included in this kit. These are multi-copy, and they allow us to address agronomic questions such as how do I get more tuber, less leaves, or the coiling, how can I change the coiling, etc. So you see the kits don't need to be just for species tree reconstruction. And what was really neat is that from the data, the sequence data, we were able to obtain not just the sequence data, but also SNPs. So, and those SNPs we used to develop a tool or a workflow, more or less. The tools were already there. So we combined a number of tools that were in the literature that allowed us to identify ploidy level of our collections, herbarium collections. And we were able to identify a cryptic diploid that was probably the parental of the more widespread polyploid of this common yam. This other kit, it's for a family of fungi. So once you have genes, it really doesn't matter if it is a plant, a fungus, or a prokaryote, or even an animal. So you have your single copy ortholog genes, 188, plus the go-to barcode, so we could integrate all of the data that is available in GenBank, NCBI. And we ended up with a massive phylogeny and revising the systematic and the nomenclature of the group. So we ended up from one genus only 10 genera. Now, in collaboration with researchers at the King Juan Carlos University in south of Madrid, we've designed yet another kit for the order Cantarellales. And what is crazy is like this topology is fully supported everywhere. It's like there's 100% everywhere. We have 500 genes. Anyways, those were like bits of a family here, a genus there, a group of fungal, whatever. So as I said, I spoke about that massive 1kp data matrix where we had 410 nuclear orthologs shared across all of Archaeplastida. So in collaboration with my colleagues in the US and folks at Kew, we designed this kit that works across all of angiosperms and targets 353 nuclear genes, single copy. Data matrix is more or less same size as the previous ones, but it has a lot more baits because we have a lot of more phylogenetic divergence. We are talking about 470,000 species across all families and orders of flowering plants. And this is yet another crazier kit. So I'm sure if you've heard of those working on prokaryotes have heard of clusters of orthologous genes, so COGs. They can be used for phylogenetic reconstruction, so tree of life, of archaea, bacteria, and a little bit, a few, let's call them protists, generally speaking. And there's a collection of 40 or so cogs that are often used for this phylogenetic inference. So we selected 10 of them and designed a kit for all prokaryotes. So this is both archaea and bacteria. And what is crazy is, like, if you do deep sequencing and you have an environmental sampling and you sequence in depth, no matter how deep you sequence, you never saturate the diversity curve. With our capture approach, you get all of the diversity that is there. We do saturate the diversity curves, and we are able to identify. We lose information on who is more abundant, but we get everyone there, which is really cool. Paper coming up in, I don't know when, eventually. So how you implement this? How am I doing with time? I'm doing well, okay. So there is a wet lab bit, and then there is the dry lab bit. Basically, DNA extraction, prepare genomic libraries, then hybridize with these kits that I've been mentioning, and then do your high-throughput sequencing. So there are a few. I mean, there's a bunch. You can go use a kit, or you can use a CTAB protocol. What is interesting is, like, when you're working with degraded DNA from collections, it's actually better to go with a hands-on CTAB approach rather than a kit, because otherwise you will not get all of the DNA that is there. And then there is this bit here that are the magnetic bits. So these things are magic. You can clean your DNA, you can size select, you can do a lot of things. They're also a nightmare to work with because they're very viscous, et cetera, and depending on what DNA you extracted or the metabolites, the secondary metabolites you have on your DNA extraction, stuff will happen. But once they work, you can get DNA out of a plant from the 1700s, no problem. Clean DNA, I mean. So then you can prepare your genomic libraries. So basically what you're doing is you fragment your DNA. If you're working with DNA from a herbarium collection, you don't need to fragment it. It's already churned into bits, like you just have tiny pieces of it. It's actually smaller than your target size, but you still can work with it. So basically you add these adapters. Then there is this bit here, which is the beads that I was showing the prior. It's actually, it has no drawings, but it's the most important bit. And here, this is just a PCR where you add indexes, dual indexes on either side. So each of your samples is individualized. And you can do this glorious thing, which is to multiplex them. So you can put all of it in one tube and sequence everything together or hybridize it first. So once you have your genomic libraries, which is basically tagged short fragments, that's when the kit that you've designed comes into play, and you have this hybridization reaction. Once you do that, you pull yet one more time, and then you sequence. So the bit that makes it HypeSeq is that when you pull here, you not only pull the enriched libraries, but you also pull the non-enriched libraries, so the step before enriching. So you put all of it together, and you sequence it. And because this is a lot of information, there's this very nice paper we published back in, I think it's 2020, where we describe the whole workflow and how to make it cheaper, okay? So I'm going to explain first the phylogenomics workflow of how now I have my raw data. So first step, I call it cut, paste, and color, which is basically you get your raw reads, and from there you end up with your target sequences. So basically you use a bunch of tools to clean your raw reads of adapters, low quality sequences, whatnot. And then you use your pipeline of choice. I very much like HiPiper, which was done by my US colleagues that I was mentioning earlier, that we both collaborated on designing that angiosperms 353 kit. So with this tool, basically you have a target file where you have the sequences that you're aiming to get and that you have hybridized for with your kit. And then you basically do mapping, and then those map reads, you put them into contigs. Those contigs are for each of your genes or targets, and then you do a quote-unquote de novo assembly for each of those targets. And then eventually you end up with your sequences. What happens here is the kits are often, because these are plants and they have very long introns, the introns can be 10,000 base pairs sometimes, I mean. So we often target just the exons on the genes, and when you sequence, you end up with the flanking regions, which is a little bit of the intronic regions. So you can have these super contigs that have both exon and intron information, which is really neat. So next step, you already have your target sequences, and you want aligned data matrices so you can do phylogenetics or phylogenomics. So this is like, like washing your clothes, a wash, rinse, and repeat cycle. So quick and dirty alignment, then a little bit of getting some statistics, balancing your data matrix, cleaning it, trimming, getting rid of outliers, and then repeating the alignments until you get data matrices that you can use for gene and species tree inference. And then, the last step for me, it's like you finally build your house, you build the wall. So you're going to have your bricks are going to be your gene trees, then the mortar, which is how you put them together, and then you end up doing this masonry bit where you end up with your species tree. So for gene tree inference, you can do exploratory trees with FastTree, or you can use HikiTree, RaxML, PhiML, so maximum likelihood for species for gene tree inference. These are the go-to tools to manipulate gene trees. And then you can use tools for species tree inference in a coalescent framework, or as it happens with plants, I said they can be polyploid, hybrid, and apomictic. You can use phylogenetic networks, but these tools are limited on how far you can, how large your data set can be and how many genes it can have. So they only go so far. So now you have an idea of why plants are cool, their genomes are crazy, what this technique is doing, so what can we do with this? So we can integrate natural history collections into biodiversity research. And I'm going to be giving you some examples of particularly this one kit, Angiosperms 353. So last year we published this capstone paper, so to speak, on Nature, where we sequenced all of the orders and families on flowering plants and compare them with the known classification. This is Angiosperm phylogeny group 4. We're now working on the 5th iteration with all of the information that we obtained in this study. It will eventually be published, so I'm guessing next year. So as you can see, there is a lot of, this is mostly done with organellar, so plastid genes, whereas this is nuclear. And there is a lot of conflict deep in time, so pertaining to the backbone, between the plastome and the nuclear history of these organisms, which is pretty cool. And now I'm gonna go from all of the angiosperms, same tool, same kit, and it's just one group. These are the umbrella trees of New Guinea. And this is us sampling in the Natural History Museum in London, these very old collections. And David Froding, he collected many of these back in the 50s in New Guinea. He was living there for a number of years. So these are all of the collections, and in white are the ones that we included in our study. So we were able to reconstruct how the group arrives into New Guinea, diversifies in situ, goes back, etc. So plants in the Wallace line, not a thing. And then this is yet another example from Southeast Asia. This one is in collaboration with local researchers and indigenous people. So indigenous people were identifying 2 species where the traditional taxonomy only saw one. So we went there and sequenced the types. And we saw that actually all of the types belong to one of the species, and that yes, there was a second one, and the names that the indigenous peoples were using corresponded to these two species, so they knew what they were talking about. So that's another really cool thing that you can do with collections and these tools. And as I said, so as you see, I'm zooming in. I'm going top down from macroevolution into microevolution. So if you get the same data set, so same raw reads into targets, now you have your sequence, you get the longest, cleanest, nicest sequence, and you use that as a pseudo-genome reference. And now you can do mapping to the pseudo-genome reference and extract SNPs with the usual GATK whatever tool, and then you can do population genomics with the same data set. So this is really good, because with one data set, you can do both of phylogeny and then implement population genomics approaches. So this paper we just published, it was done in collaboration with Manitza, or led by Manitza Ballant, who just defended her PhD a few months ago. Excellent researcher based in Barcelona. And we got collections from the entire known natural distribution of cannabis, mostly from herbarium collections from Mongolia, Iran, and places you don't often go to, let's say it that way, or cannot go to. And we were able to, I mean, it had a lot of geographic structure, regardless of whether the plant was used for fiber or recreational purposes or whatnot, okay? And again, we got both the phylogeny and the population genomics analysis that were telling complementary stories. This paper we're now reviewing, it's under review. And it's Genus Thymus in collaboration with Regina Berjano, a professor at the Universidad de Sevilla. And same, so we got our phylogeny, and then we have our population genomic studies. And it's crazy, because this yellow beet used to be included with the blue beet, and they were considered one species, and this one was a subspecies of this one. Turns out all of this is one species, and all of that it's another. This is a tetraploid, all of these are diploid. And there is gene flow between ploidy levels. We don't know whether this gene flow between ploidy levels, so it's mediated by triploid bridges or what other, but it's crazy that we're now able to see these things happen with collections or with fresh material. This case is fresh material. This is mostly collections. And that's it. Thank you.
Thank you, Lisa. So we have like 15 minutes for questions.
Hi, well, thank you very much. I found very interesting the talk. And I was wondering, I found very interesting this map you were showing about these euphorbias. In which you found a similarity, between the Canary Islands and the Arabian Peninsula, that this hypothesis for the rig vegetation that I heard.
It's called a run flora.
Run flora.
Yeah, it's the run flora pattern, and there's a bunch of lineages that do this.
Okay, so have you been able to date the split? I mean,
Yeah.
because apparently all this happened, when the Sahara Desert dried up, because at some point they were together somewhere where today you have this desert. I don't know if you have been able to date it and if it matches what you will be expecting according to the climatic changes and so on?
Yes, we published this back in 2015. We actually were able to see it just with Sanger data. So we had 14 lineages across flowering plants that shared this pattern. They also can reach South Africa and Namibia, so Southern Africa. So RAN is German for edge or border, so the edge of the continent, margins of the continent and the archipelagos that are around of it. That's where you find these lineages. So these lineages are neither too xeric nor too, they are not too tropical and not too xeric. They also live on the edge of these two climate streams. And what we were able to show is that depending on their tolerance for aridification or tropicalization, whether they could tolerate a more tropical or more arid environment, they would diverge closer to present. So the more aridity they could tolerate, the later they would diverge. And the more tropical they were, the older the divergence was. So this is what we call a pseudo-biogeographic congruence example. And yeah, the paper is out.
Very interesting, this last point you have pointed out. Thank you very much.
Yeah.
Any other question for Lisa?
Hi, Lisa. Thanks for your talk, first of all. I've seen in several of your papers you have tackled time-calibrated trees. Was your preferred approach for such datasets?
Yeah, so that's a good point. As I briefly mentioned that, so plants have reticulated topologies, and that the phylogenetic network methods only go so far. So for time calibration, when you have very large data matrices, the Bayesian methods only go so far as well. So there's a limitation on the size of your data matrix. So my go-to approach when you have a not too big genome would be Bayesian, BEAST, most probably, 1 or 2, pick your poison. But when you're working with these very large data matrices, I do exploratory time-calibrated trees because I don't really trust the inference fully. So I use penalized likelihood. So there is a tool called 3PL. I also use RelTime, which is implemented in PAML. And with those two tools, I can get a very good idea. PAML allows you to incorporate a little bit of uncertainty on your estimates, whereas with 3PL, it's very categorical. You sort of have a point estimate, and then you can bootstrap and get a little bit of error around that point estimate. But it's penalized likelihood. It only goes so far. It's very... So when you go, if you want to do a more Bayesian-like approach, there is MNCN-CSIC, which is implemented in PAML? Yeah, PAML. It's part of PAML. Zihen Zhang, I think it's the main author in collaboration with Mario dos Reis. So basically, you have a fixed topology. So you have to trust your species tree. And then from there, you can go fully Bayesian. It takes time and a lot of trials, but that's the most close approach to what we used to do with BEAST. Another approach which also works, it's what is called gene shopping. Rather than working with your entire data matrix, you select genes, but you don't select the ones you want or them randomly. You actually need to know your data sets, so you use a number of tools to calculate root to tip variation, whether your tree is congruent with your species tree, because one of the things that happens often with gene tree, species tree is that there is a lot of conflict, and your gene tree may be telling a completely different story than your species tree. So, so once you've shopped for those genes that meet a number of requirements, there are a few tools out there. There is Sortodate, and there is gene sorter, I think it is, or something like that. I don't remember right now off the top of my head. With these tools, you can shop for, let's say you have a data matrix of 300 genes, you just end up with 25. And you also have often to shop your in your tips. You know, like you cannot get all of the tips. You get the most relevant ones, so you can have all your time constraints and the crowns and stems that you need in that topology. And with that, then you can use BEAST. So the phylogeny that I was showing here, this is BEAST. But it is using 25 genes out of the couple hundred we were able to get from these very old herbarium collections. And then we were able to do the full BEAST dating inference. Okay?
Yeah, great. Great. Good to know that you still trust concatenation plus penalized likelihood, because I read some people who really didn't really agree, but I trust you. And also, gene shopping is a nice approach too. Thank you.
No problem.
We have time for one last question.
Hi, thanks. That was great. I have a question. So, when you present all the different probe kits, right? And you talk about 600 genes or something like this, and that you can use them at different kind of scales of divergence, right? So how do you find the differences in phylogenetic information content between your loci, and how do you deal with that? So there are a number of tools out there that you can use to calculate phyloinformativeness, or how many parsimony informative characters you have in a given alignment. So basically, I align my genes, and then I calculate the statistics, and I can see how informative or not they are. So what I look for when I design this kit is that all of the genes are balanced, that there are not a few genes that have all of the information. So they need to be, if they're going to be for phylogenomics, so a species tree inference, I need them to be single copy nuclear orthologs. So once I have these single copy nuclear orthologs, I don't want to have very short ones and very long ones. I want to have average size ones. So let's say 1,000 base pairs medium size. And then I don't want to have the longer genes having all of the information, or the short genes having no information. I mean, if it has no information, just don't put it in the matrix, because the baits are expensive. So using, for instance, a mass, it's by Borovich. He works on ants. He has this very cool tool. And one of the things is that it calculates summary statistics of your alignments. And I use those summary statistics to inform the selection of what's going to go into the target capture kit. So it's not random, it's informed on. And so once I have selected whatever is actually in this figure here, here. See, you can see here all of the filters that we're applying. So we have a first filter where we remove multi-copy. Then we will have missing data when we're designing this, because we have sequences of different oranges. Sometimes there are transcriptomes, assemblies, genomes, whatever is in NCBI or that we have generated ourselves. So we want our data matrix to be as complete as possible. And then, like all the uninformative genes, we kick them out. And then here,
You know, like see the length outliers, whatever is on the edges of the range of all the genes, they also go out. And then here we include, for instance, the usual barcodes. And lastly, you know, like we make sure that we have the right size. So we will even kick further out genes to make sure that our kit is not too expensive. Because as I said, depending on how many baits, the more expensive. So the baits can be 120 nucleotides, but they can also be 80 nucleotides. There is a tendency now to go to 80 nucleotides for the captures, mostly because when you start working with environmental DNA or collections, herbarium collections, etc., you will not have fragments that are 120 base pairs. Most of your fragments will actually be 80. So after all of these filters, so you know, like there is a long way until you get your data matrix, and then there is a lot of filtering until you end up with your kit. So you do all of this filtering until you end up with a kit that you have all of this information. So you know what genes, how ortholog you are. You have a prior phylogeny. You know how well it works. So this is the design of the kit. This is a phylogeny that we did after we selected all of these markers, and it works. So we know before designing the kit that we're going to have a 100% supported topology for this group, using multi-coalescent concatenation approaches, etc. So the really neat thing about this is that I don't have to suffer figuring out who is the ortholog. I go in there already knowing that. I just have to go and do the reconstruction, phylogenomic or population genomic reconstruction. So that's very neat.
Thank you very much, Lisa. So we're going to move on to the next speaker. Thank you, Lisa.
Thank you.
Cool. The next talk is by Hannes Svardal. He's associate professor at the University of Antwerp in Belgium. He has a bit of an unusual path study-wise. He has been combining mathematics, physics, and biology. He did a PhD with Joachim Hermisson at the Institute of Mathematics at the University of Vienna, and then a postdoc at the Wellcome Sanger Institute in Cambridge, UK, working with Richard Darwin, among other postdocs and so on. He's very well known for his contributions to understanding the genomic basis of explosive radiation in cichlid fish. Again, he has done many things, but that's the thing that I knew. And today he will talk about applications of genomics to studying microevolutionary processes that give rise to new species. Thank you.
Yeah, hello. Good morning, everyone. Can you hear me well? Is it good? Okay. Yeah, great to be here. So I was asked to talk about my work and to also give a bit of conceptual background and talk a bit about methods. I hope that I find a good balance, but I stay rather high level. So if something is unclear, please feel free to ask afterwards or send me an email. So I guess the reason that we are here is because most of us are interested in the amazing biodiversity out there, and we are trying to understand it, which might help us to predict it and to protect it. Overall, as natural scientists, of course, nature out there are some processes which lead to a pattern. And as natural scientists, we go the other way around. We kind of see the patterns, we observe the patterns, especially, of course, in a natural history museum is a perfect place for this. And we use these patterns to actually try to understand the process behind them. Because if we understand the processes, then we can actually go further than just understand this single pattern, but we can try to interpolate to other patterns to get a more broad understanding of nature and potentially to kind of predict what happens if we change the boundary conditions of this process. So my own background, my own research interest is really in the evolutionary mechanisms behind the evolution of biodiversity. And to do this, on the one hand, we use computational methods for genomic inference. On the other hand, we work on kind of trying to understand the molecular basis of phenotypes, because of course, phenotypes are the link between the evolutionary pressures, which kind of select on phenotypes, and on the other hand, the DNA, where kind of the changes happen and are inherited to the next generation. In the background, you see some of the species that I've been working on. Today, I will really focus on cichlid fish. So I have to say this is a computer-generated image, so don't take the pictures at face value. But they should give you an idea. I showed you this picture of biodiversity, but of course, these are actually not unconnected things, but in reality, they all are connected to each other because they stem from a common ancestor. And that's really, for me, one of the most mind-blowing things about evolution, that there is actually this process of speciation where we get lineages that diverge, and all this diversity can be traced to a common ancestor, also in terms of DNA sequence in the end. And this process is really what I would like to understand. And of course, it's also kind of the question which motivated Darwin for his work. And if today we have two species, then of course there is some kind of past to it. There was some ancestral species, and there was a process which caused them to become separate species. And this is what we are trying to understand. Classically, Ernst Mayr and this kind of school of thought, there was a strong focus on geographic isolation, so that you have some kind of barrier, and then things on either side of the barrier just start to become different over time. But I think, especially now in the age of genome sequencing, it becomes more and more clear that very often you can get what's called ecological speciation, so where there are different environments and there is selection to thrive in these different environments. And this can actually lead one ancestral species to start diverging, first kind of getting different locally adapted populations, and then at some point they also evolve some reproductive isolation to actually become separate. And this is really this ecological speciation that we mostly work on. So how can this actually happen? How can you get separation despite incomplete reproductive isolation, and how can reproductive isolation evolve as a kind of following selection pressures for kind of staying different? And to study this, we work mainly with genome sequencing data. And more specifically, actually, we are focusing on a lot on hybridization. This is the question of how does this divergence happen? And it seems that if there is hybridization from a third lineage, or if there is actually hybridization in this ancestral lineage, this is something that can actually kind of help the process of speciation, and we're studying a lot the effect of this. And I'm going to give a bit more background on this. But first, I want to define the term of evolutionary radiations or adaptive radiations. This is the idea that speciation is often not just a thing where we get kind of two lineages involved, but often we get these very rapid bursts of evolution. And one example is, for example, the stem lineages of birds and vertebrate, birds and mammals. At the time when dinosaurs went extinct, there were probably just many ecological niches left empty, and these were quickly filled, and you really kind of see this surge in evolutionary lineage in a relatively short amount of time. And one important question is, how can this happen so quickly? How can you get so many species evolving so quickly? And of course, Darwin finches are a classical example for this. And one problem is that actually mutation is a very slow process. So on average, you only have around 10 to the minus 8 de novo mutations each generation, depending on the species, but something around that order. But of course, to adapt to different ecological niches, it's usually not one mutation that's sufficient. Usually you kind of need many different adaptations. And one thing that's, as I told you, is kind of found more and more frequently is that hybridization between divergent lineages can play an important role. And the reason is that you basically can get combination of different genetic material and can form something new, not needing to wait for mutations to happen. And what's really important, of course, is recombination here, because you kind of dehybridize, but then they must backcross or kind of mate with each other. And recombination is the process which then allows kind of the combination of different alleles at different genetic loci. But there is a kind of conflict of interest, because on the one hand, here recombination is good because you can kind of combine different ancestry. On the other hand, on the other side here, when they actually start to diverge into different ecotypes, they shouldn't kind of hybridize anymore, because then they will just kind of create unfit intermediates, and it will kind of impede them from diverging if they hybridize. So kind of at this side recombination is good, and on the other side recombination actually between lineages can erode the adaptation of the different species. So, but within species, recombination can still be good because it's a process that can help to purge deleterious variants. So how can nature actually resolve this antagonism between kind of the role of the kind of the favorable role of recombination for speciation and the impeding role? So one possible mechanism are chromosomal inversions. A chromosomal inversion is a type of large-scale structural rearrangement or large-scale mutation where a piece of DNA flips end to end. And one thing about these chromosomal inversions is that often they have relatively mild phenotypic consequences, because you still have the same genes, the large-scale gene order is still conserved. If there are kind of 100 genes in the inversion, they're still next to each other, these 100 genes. It's just the overall block that has changed its orientation. So often they don't have very strong phenotypes on their own. But one kind of very important effect is that this suppress recombination in heterozygotes. So if there is an individual which has kind of one copy that's inverted and one copy that's non-inverted, then recombination doesn't work during meiosis. However, oops, that was the wrong button, yes. However, if you're homozygous for the same orientation, then recombination just works fine. And it's found more and more in different systems that inversions play an important role in local adaptation. They're also called supergenes often, because you can get kind of consistent adaptation in different genes which are in this block, and they cannot be broken up by recombination. And this is very important, especially for complex phenotypes, which often have a complex genetic basis. And of course, sex chromosomes are a prime example of inversions, where you get then suppression of recombination between the different sex chromosomes. Our main model system are, I'll step here. I feel like I'm making lots of noise when I'm walking. So is this okay with the camera? Or I don't know whether I'm filmed now. No, it's okay. Okay, good. So African cichlids are our main model system, and they're a great model system because they have these amazing adaptive radiations in different lakes in Africa. So in Lake Victoria, Lake Tanganyika, and Lake Malawi, you have these big adaptive radiations where one or few ancestral species came into the lake, and then they diversified into many, many different species in situ in these lakes. And what's striking about this in Lake Malawi is, if you think again about this kind of vertebrate tree of life that I've shown you before, then all these 800 species basically emerge at the very, very tip of this tree. So it's kind of a really evolutionary, very fast time. So that makes them a very good model to study this rapid evolution of biodiversity. And yeah, here you can see it's a kind of a very big radiation in a very short amount of time. It's actually, I think it's the largest recent vertebrate radiation in terms of extant species. And despite kind of this rapid pace of this radiation, you get an amazing amount of phenotypic diversity. I need to credit my postdoc Valja for making these very nice pictograms of different species. Actually, here you see them a bit more to scale. You see that some of them are really tiny. These are all adults. And some of them are kind of huge predatory species. And this is kind of the first results slide. So what you see here, you can't read the axis, but this is in gray, the genetic diversity, so heterozygosity within species, and in orange, the genetic divergence between species. And the first thing you can see is that these distributions are totally overlapping. So the divergence between species is really at the same order of magnitude as the diversity within species. And this divergence is 10 times less than humans are separated from their closest relative, the chimpanzee. So all these 800 species kind of 10 times less divergent than human chimpanzee. So the first question we wanted to address is whether hybridization is playing a role in this adaptive radiation. And there are two ways that hybridization could play a role. One is that as this radiation unfolds, there could be hybridization between different species, and this could either help or impede the radiation. And on the other hand, ancestral to the radiation, you could have some ancestral hybridization events, which kind of create kind of a hybrid ancestor. And this is what is called an ancestral hybrid swarm. So we kind of wanted to look for both of these patterns in Malawi cichlids. And the main tool we used for this is what's called Baba tests. This is a common statistic, which I think many of you will have heard of. And the basic idea behind it is that when you look at a phylogeny that looks like this, but let's say it's kind of not a true phylogeny in the sense that there was some introgression from this lineage C into lineage B of a certain percentage. Then what happens is, of course, if you kind of assume a pure phylogeny, A and B find a common ancestor with each other before they find one with C. So they should be equally closely related to C. But if there is a certain percentage of introgression from C, then you expect that B is a bit closer to C than A is. And basically, this is the whole logic behind these Baba tests or D statistics or Patterson's D. So there's a whole kind of statistical machinery behind this, but it's in principle relatively straightforward. And the cool thing is that it's very powerful to look for these patterns in genome sequencing data. And these patterns have actually revealed that almost anywhere you look, plants, animals, you find tons of hybridization between species. So it seems that it's really kind of the rule rather than the exception that this happens. Not necessarily between extant species, so it doesn't mean that you kind of see hybrids out there, but within their evolutionary history, within this kind of hundreds of thousands or millions of years, these things happen. And to make sense of this, for Malawi cichlids, we had a very large data set of whole genome sequencing. So for this study, it was like 96 species. So we developed kind of some summary to summarize these many different comparisons where you use these trios, as you have seen on the slide before, to actually see kind of the major patterns of genetic exchange between species. So I'm not going to go into detail, but basically the red means that there's evidence that there was gene flow between these clades. So there's on the x-axis and the y-axis is the same phylogenetic tree. And we could also kind of see when it happened with specific ancestral branches. Applying this to Lake Malawi cichlids, basically the conclusion is that there is lots of hybridization. It's not happening all the time that we can observe them out there, but on evolutionary timescales, this is a very frequent pattern, that there is exchange between the lineages. So now about the second question, about an ancestral hybridization event, or this kind of hybrid swarm scenario, which is something that kind of Ole Seehausen has kind of shown for Lake Victoria and has also kind of talked about a lot. And actually, looking for such a pattern, indeed, this is what we found for Lake Malawi as well. So we had some outgroup lineages, and basically we could show that Lake Malawi cichlids are basically a 75 to 25% mixture of some extinct lineages which are related to these outgroup lineages. And we could also show that this hybridization-derived polymorphism actually helped the species divergence. So this kind of was under selection during adaptive radiation in these cichlids. Okay, now remember the picture I showed you before with the washing machine and the birds. So we said there is this kind of conflict of interest with gene flow and recombination. On the one hand, kind of it's good for creating diversity. On the other hand, it might be bad for actually completing speciation or kind of maintaining species differences. And I showed the potential solution of inversions. So the question is, do we actually find such inversions in Malawi cichlids? And to address this question, we did a very comprehensive sampling of Lake Malawi. So we collected lots of specimens, and we produced short-read whole genome sequencing. So I should say most of this was done in the lab of Richard Durbin when I was postdoc there, of the sequencing. And yeah, so it's whole genome short-read sequencing of 240 species, 1,375 individuals. Yeah, if you have questions about the details, I'm happy to answer them later. But first, let's get to the question, how can we actually detect chromosomal inversions? So the classical approach is cytogenetics, but the problem is that cytogenetics is very good at seeing kind of things like chromosome fissions or fusions or major events where you really see a karyotype change, but with inversions, most of the time you don't see a karyotype change. So what you would actually need is you would need some fluorescent probes on the right places on the chromosomes so they can actually see what's happening. But of course, for this, you first need to know where these inversions are. So that's why this was not the method of choice. Another thing you could do is long-read sequencing. And the idea behind this is that when you have your long reads and you align them to the reference genome, they would just kind of align. But imagine that you have a genome which is kind of inverted relative to your reference. Then actually what you're going to sequence are kind of reads which kind of have chunks which are not next to each other in the reference genome. And if you were to align these reads to the reference genome, what you would get is split alignment. So basically, the left-hand side of your read would align somewhere in the genome, and the right-hand side would align in a flipped way somewhere far away. These split alignments would be a typical sign for inversions. However, there are two problems with this approach. One of them is we didn't have long-read sequencing, we just had short-read sequencing at the time. And the other one is that these inversion breakpoints are often very repetitive. So the reasons that inversions occur in the first place is often because there are 100 kilobases of tandem repeats on either side, and then it flips. And if you have 100 kilobases of tandem repeats, your long reads are still only 10 kilobases, 50 kilobases, so it's very challenging still to kind of map over these breakpoints or even assemble over these breakpoints. Now it's getting better now that we are close to telomere assemblies, but overall with kind of regular short reads, long reads, it can still be challenging. So now the question is, how about short reads? Can we kind of find inversions with short reads? And this kind of approach of kind of spanning, since I told you this kind of spanning a breakpoint is really challenging with long reads, so there is not much chance with short reads, even with kind of read pairs. So, but however, there is another pattern. Of course, what do inversions do? We said kind of the main effect is to suppress recombination. And of course, recombination is a very important process in evolution. So actually, the fact that they suppress recombination can leave a population genetic pattern in your population. And it's this population genetic pattern that we were really looking for. The first potential signature is related to phylogenetic discordance. So this is this effect when the gene tree is different from the species tree, also called incomplete lineage sorting. This is something that regularly happens when you have closely related species. But what usually happens is, since kind of as you go back into the past and go through different ancestors, recombination will make different parts of the genome independent. So incomplete lineage sorting will usually have a relatively short scale that you kind of get gene trees changing relatively quickly along the genome. And if you make an average, you soon get to something that's close to the species tree. However, in inversions, this is of course different because there is no recombination. So then actually you can get long stretches where you consistently get a phylogeny which is different from the genome-wide phylogeny. And this, of course, is especially true if these inversions segregate across species. So if the inversions are older than your species and have been polymorphic along these branches and possibly have been introgressing through hybridization. So this is the first difference. And of course, another important process in evolution is selection. And again, it's the same that selection has a local signature which kind of extends due to linkage, but as you go away, recombination will kind of make regions independent, and you see less and less the pattern of selection. But if there is inversion, again, the signal can have a much larger effect, which could lead to kind of reduced diversity across kind of a longer part of the genome. So in summary, there are population genetic signals that can make that inversion regions look kind of different in not so much both kind of in the overall pattern compared to the rest of the genome, but more in the scale, so that the change of the pattern kind of stays more consistent across a large region. So how to test for these kind of patterns? Basically looking for regions which have an evolutionary history consistently different from the rest of the genome. And before I kind of tell you how we did this, I want to give a bit of background on Malawi cichlids, because this will be important for interpreting the results. So Malawi cichlids have these 7 major clades, which are 2 pelagic clades, one mainly predators, Rhamphochromys, and one mainly deep water pelagic planktivores, Dipotoxodon. Then there is this very diverse clade of benthic species, which are all very closely related to each other, but we can distinguish a clade of shallow benthic species and a clade of deep benthic species. And the clade of mainly pelagic planktivore Utaka. Then the most famous Malawi cichlids are down here. These are the rock dwellers. They're also called Mbuna. If you see people that have Malawi cichlids in their aquaria, this is mostly Mbuna because they are kind of very colorful and also quite aggressive, so you can see them displaying and fighting. And then there is a riverine lineage called Astatylabecaliptra, which interestingly is kind of very derived. It sits inside the radiation, but actually it retains kind of an ancestral phenotype. So if you go to the rivers and sample outgroup species, they all kind of look like Calyptra, just because they have this kind of riverine phenotype. So you have one of those within the radiation as well. And probably kind of each of the ancestors looked a bit like this. This is kind of our hypothesis that we proposed in another paper. Okay, so these are the major clades. And now what I show you here is again the same major clades, only it's a bit flipped, sorry. These are the pelagic, these are the rock dwellers, and these are the benthics. And you can already see these benthics are all kind of closely related. So what you see here is a pairwise distance matrix. So this is just kind of aligning the genomes and then counting the SNP difference between different species. And you count how many differences do they have. And the more red this is, the less differences they have. So you see within groups, they kind of have less differences, which makes sense. And between groups, they have more differences. And so this is for the whole genome. But in principle, we can do the same thing for parts of the genome. So we do this here in windows along the genome. Then we cluster these with kind of reduced dimensions with a principal component analysis in these windows. And then we cluster them with a clustering algorithm. And yeah, I'm not asking you kind of to understand or look at all this, but basically on the x-axis are the chromosomes here. On the y-axis are different numbers of clusters. And the colors say which cluster a region belongs to. And the only thing important here is that there are some regions which kind of show this consistent pattern of looking consistently different, having consistently the same color, so that they kind of have consistently an evolutionary history which is different from the kind of genome-wide average. And we can summarize this in what we call cluster density score, so that you kind of see regions in the genome that show this pattern. And our hypothesis was that these regions correspond to chromosomal inversions. There are some other ways to look at this. One of them is a standard principal component analysis, which you might know and have seen in the last presentation, only that instead of plotting two principal components axes against each other, you can just take the first principal components, which is the major axis of genetic variation, and plot it against genomic position. So again, this is principal component windows. And when you do this along one chromosome here, for example, this is what you would expect, that kind of you get the different colors of different clades, so the clades are different from each other. But then actually in this region, you get these 3 bands which don't correspond to the clades, but to something else. And this very much looks like 3 genotypes of an inversion, where you get the homozygotes on the edges and the heterozygotes in the middle. And indeed, when you look at the diversity within these groups, so the heterozygosity, you see that the middle clade has a much higher diversity. So it's consistent with this being a heterozygote for an inversion. This is the summary of all chromosomes of this principal component analysis. And the summary here is that we basically discovered 5 very large chromosomal inversions, which are more than half a chromosome in size. And there are some smaller ones which don't come out so clearly and which I will not talk about today. Now that we kind of know this, we could actually design probes for fluorescent in situ hybridization, and we could indeed see that these markers kind of jump here, and they don't jump in a way that is expected for a single inversion, but actually jump into the middle, which suggests there is a double inversion. And this is also what we found with long reads. We found for this chromosome a double inversion. The other chromosomes were kind of a single inversion. Then what we also did, or with collaborators, is to look at recombination in hybrids. So of course the important effect is that they do suppress recombination. We showed this on an evolutionary scale, but can we actually see this also in more in a direct scale of a pedigree? And indeed that's what we find. So this is again such a PCA along a chromosome. Now the 2 extreme bands are the 2 parental species. The F1s would be exactly in the middle, and then F2s and F3s, you would expect them to kind of move between these bands wherever they have a recombination event. And we see this in the flanks of the inversions, but inside the inversion there is kind of no recombination at all. Now, to look at the phylogenetic distribution of these inversions, I first plot the phylogeny here, and what I did here is only the benthic clade is expanded, because you will see the benthics are kind of the place where things are happening, and the other clades are here. So here are the deep benthics in blue, the shallow benthics in orange, and the Utaka are not monophyletic in this group, but they are here and here in this tree. Now, if I add the inversion states to this, it looks like this. So each of these bands is the frequency of the inversions in this group. If it's white, this means it's absent. And what we see is that these 3 outer bands, the inversions of chromosome 10, 13, and 2, they are mostly present in this deep benthic group. So they are present in most deep benthics except for this one clade, which actually lives in the shallow waters, despite genetically belonging to the deep benthic group. But then we also see that they appear in some parts of the shallow benthic tree. Then the other 2 inversions, the one on chromosome 11 in red, this is actually present in a group that contains deep benthics and some shallow benthics, and also in this pelagic diplotoxodon. And the interesting thing here is that it's actually a monophyletic group which mostly has the ancestral non-inverted state in the benthics. This is actually not what you would expect. When you expect an inversion appears and then fixes, you would expect as a monophyletic group which has the derived state. But here it's the opposite. It's a monophyletic group which has the ancestral state. Chromosome 9, on the other hand, is basically present in all benthics and in diploxodon. But what's really puzzling about chromosome 9 is it's actually heterozygous in several species of benthics, in many species, but we never ever found it present in homozygous non-inverted state. This state didn't exist. So these populations where it's polymorphic, it's only individuals fixed for the inversion or heterozygous. Okay, so we wanted to understand the evolutionary history of these inversions given these patterns, and of course, ideally one would do some fancy modeling to try to understand this, but actually the patterns were really confusing for many reasons, and in the end, what we mainly focused on is phylogenetic relationships in the inversions. So what I show you here is a tree that's built for outside the inversion. So this is not how the inversion haplotypes are related, but how the species are related. Of course, I could also build a tree in the inversion region and look at this. And this is also what we did. I'm not showing this, but we kind of built trees for all these inversions and then kind of tried to compare the trees and try to reconcile them with an evolutionary history. And on the other hand, we just looked at divergence patterns between these different groups for non-inverted regions and for the different inversion haplotypes. And I'm not really asking you to follow these plots because I think it's very complex and it took me a long time to really understand what's going on. And each time I look at them again, I still, again, don't understand what's going on. But just two features that are interesting. One of them is, so this here is the ancestral haplotype and how it's related to the different other groups. And the puzzling thing is that it's actually the ancestral haplotype for benthic. So the benthic ancestral haplotype is much more divergent, so this is divergence time on this axis, is much more divergent from all other groups than any other inversion haplotype or than the rest of the genome. So it looks like the non-inverted haplotype in benthics is a very ancient haplotype which is not present anywhere else. And it's actually the inverted haplotype which is more close to the rest of the groups, although they don't have the inversion. So that's kind of one interesting pattern. Another interesting pattern is that there is actually another group, which is Astatia calyptra, which is unexpectedly close to the benthics given this tree. You don't expect calyptra to be closer than the Mbuna, for example. And basically, looking at these patterns for a very long time and trying to understand what's going on, so a very long time, we came up with this simplified evolutionary model of what's happening. So these are the different major clays that I showed before, the pelagics, then the benthics in the middle, this riverine and the rock dwellers. And I told you that here is an ancestral hybridization event that seeded the radiation. But actually what we found is that there was an additional hybridization event that seeded the benthic lineage between this pelagic Diplodoxodon and this riverine calyptra. And then the different colors are the different inversions again. One inversion is shared with Diplodoxodon, two inversions are shared with them. And the reason that we had this strange phylogenetic pattern that there is the ancestral haplotype, which actually is kind of monophyletic, is because it reintrogressed. So probably the benthics were fixed ancestrally for the inversion, but then there was a reintrogression of this haplotype. And not only this chromosome 11 haplotype, but this chromosome 9 haplotype, which is never homozygous, outside of the radiation, from a lineage which is not part of the Malawi radiation. So that's why it's such an old haplotype. And we could also kind of point to a probable lineage that it came from. And then we see that within there is kind of lots of exchange between these haplotypes, between the lineages, and we think that this contributes to adaptation. Okay, so I think I need to speed up a bit, but what we did next, of course, we are interested in was there selection, what kind of drove the evolution of these haplotypes. And in short, what we did is we focused on mutations that happened very early on these haplotypes, because those are probably the ones that drove their evolution. And to identify these mutations, we basically looked at mutations or SNPs for which the genotype is strongly correlated with the inversion genotype. And you can plot this along the chromosomes, and you see, of course, the highest scores are inside the inversions. It's, of course, lower outside the inversions. And an interesting feature of these SNPs, so this is the correlation coefficient here. So the SNPs with a high correlation coefficient are kind of either at 1 or at -1. We found that these SNPs are kind of more likely to be in exons than SNPs which are not strongly correlated with the inversions, which could be a sign of positive selection or of relaxed purifying selection. Oops. But when you then actually look at the DNDS ratio of these SNPs, so again, this is the correlation coefficient, and if you look at the ones which are most strongly correlated with the inversion, they show a strong enrichment for non-synonymous substitutions, and the ratio is actually above 1. And the ratio above 1 you can only explain with positive selection. So we do think that these kind of mutations that drove the early evolution of these haplotypes are enriched for positive selection. And of course, then we wanted to know kind of what genes were involved in this. And the classical thing that people do is a gene ontology analysis. But the problem with this is that most of the gene ontology annotations actually come from human or mouse or so. They are actually not very specific to fish. So to do something that's a bit more specific, what we did is we looked at single cell expression in different tissues of developing zebrafish. Again, it's a relatively divergent species, but closer than human. And what we found is that there is a strong enrichment in different sensory tissues and the nervous system. So there's an enrichment in different inversions for photoreception, mechanoreception, and chemoreception. And this makes sense. And also if you look at the genes, so there are lots of opsin genes, and there are genes which are kind of known to be involved in different functions like this. And this is interesting on the one hand because, of course, sensory systems are very important for foraging and for ecological adaptation. But on the other hand, they are important for mate choice as well. And this mate choice is the main factor that keeps these fish apart. So there are candidate genes. I think I need to speed up a bit, so I will go a bit faster or skip some parts. So then, of course, the next thing is we found this enrichment and selection, but what are the phenotypes? And this is kind of a tricky question because within each of these groups, there is so much diversity that it's not obvious to say, you know, the inversion makes the fish blue, because within the inverted and non-inverted state, there are blue fish, red fish, green fish, yellow fish, and so on. So there is kind of lots of diversity within the groups, which could either mean that it's more general phenotypes, or it could mean that the inversion haplotype actually has species-specific functions. So there are other mutations which are important, which happened later within species carrying these haplotypes. But one pattern that is quite clear is that depth adaptation might play a role. Again, these sensory systems we saw, they of course are important for depth adaptation, but also we see this strong correlation of inversion state with depths, and there is some phylogenetic confounding in this, but also within the shallow benthic, there are some species which live deep, and those tend to have the inverted haplotypes, and vice versa. It's hard to say what is cause and consequence, but yeah, our kind of best hypothesis is that there is a lot of depth adaptation going on these haplotypes. One potential thing could be these sensory pores. So some of these deep-living species have these large sensory pores, and we do find outliers for the presence of these sensory pores in the inverted regions. About depth adaptation, I just wanted to give one plug on. It's actually a different study, also on Malawi cichlid, specific on depth adaptation from my PhD student, Julia. It was just published. And she was kind of really focusing on depth adaptation, not specifically about inversions, but generally genome-wide, focusing on this clade of Diplotaxodon, this deep water pelagic species. And she did different approaches, including a genome-wide association study, where you try to associate genotype with phenotype, different selection scans, and differential expression analysis. And this kind of showed a relatively clear pattern that there was strong signals of adaptation in this phototransduction cascade. So basically, almost every gene in this phototransduction cascade was basically found as an outlier by more than one of these methods. And these methods really kind of look at independent signals in the data. So this really kind of gave an impression that yeah, this phototransduction is kind of a major pathway of depth adaptation. Other results included also hemoglobin and different kind of hypoxia-related categories, which also make sense because oxygen saturation and pressure is, of course, also different at the depths. Another kind of cool potential phenotype is bower building. So bowers are these mating platforms that males build, which are really important in mate choice and assortative mating. And it turns out that previous studies actually found a strong correlation of kind of this power shape with these inversions. So actually, this is the chromosome 11 inversion, this double inversion that I told you, and on chromosome 2 is also an inversion. So the problem is that this study really was phylogenetically confounded, so it's not very conclusive that there really is an association with power building. So we are now working on kind of designing more specific within population studies to address this. Finally, the one very clear phenotype that we found is that actually in some populations, these inversions act as sex chromosomes. For example, in this species, Copolychromys chrysonotus, males are always heterozygous, while females are homozygous for the inverted state. And we could confirm this in kind of lab stocks of different species. And very interestingly, in Otopharynx heterostigma, actually different populations use different inversions as sex chromosome. And this is chromosome 9, which is the one which we never found homozygous. So we think it actually might be a sex chromosome in many more species. And that's why it's never homozygous, and basically there's no double Ys. However, our sample was kind of such that for most species we only had kind of 1 or 2 samples, so we can't really map sex within the species with high confidence. Also, sex determination is tricky in cichlids. The last thing to mention is that one kind of very interesting feature of this sex determination is, so overall we find for these 3 chromosomes an association with sex determination, but you can see it's by no means perfect. So it seems that it's very variable and changes very quickly. So in some species it's sex determining the inversion, in some it is not, and it seems to change. But one general pattern is that in each case where we found it to be sex determining, it's actually that the non-inverted haplotype reintrogressed in this population. So for chromosome 9, chromosome 11, and chromosome 10, it's always in cases where the non-inverted haplotype reintrogresses in a population that has been fixed for the inverted, where suddenly the non-inverted becomes a Y chromosome. So this is quite interesting, and it's something that I have not seen in other systems. We have some hypothesis why this happens. And it's, I think, interesting to follow up. So it's the reintrogressed non-inverted haplotype that takes up this Y-like function. So to conclude, we found these 5 large inversions. They kind of helped us to reconstruct a simplified but still quite complicated evolutionary history of these lineages. It seems that there is a relation with depth adaptation. We found there was positive selection on different sensory systems, and they are involved in sex determination in some species, but not in others. So the open questions really are, what really happens during speciation, because this was more like a large-scale picture that we drew, but we didn't really kind of look at individual speciation events and whether these inversions played a role there. And of course, to pin down the phenotypes, because now we have kind of some ideas, but we don't really have that's what the inversion does in this context. And then, given that they contribute to sex determination and that they have these sensory systems which might play a role in both natural and sexual selection, we wonder whether there is some kind of interesting interaction going on. So this is what we want to follow up. And of course, we now found these very large inversions, but from other kind of preliminary results, we think that that this is only the tip of the iceberg, and there are kind of many more smaller inversions and other structural variants which affect recombination and play an important role in this diversification. And this is another thing we would like to follow up. So if you want to read up on this, the inversion stuff is published, and depth adaptation is here, and there's also a bit more on, even kind of a more complex hybrid swarm history that I didn't talk about today with other lineages contributing. Okay, with this I thank you for your attention, and of course I thank all my collaborators. Some of them are shown here, but not all. This was a very large collaborative study. So for example, Eliano, who is present here, was also involved in this. So thank you for your attention, and I'm happy to take questions.
Thanks, Hannes. Super interesting talk. We have time for some questions now.
Thank you very much for this very nice talk. I was wondering, you said At the very end, you said that you would also like to look at smaller things, because during the talk I was already wondering what is the size range basically that you're looking at? More or less whole chromosome inversions, or?
So the ones we worked on, they were half a chromosome, which means like 10 megabase, so between 10 and 15 megabase. But yeah, we know that there are some smaller ones. I mean, if you look at whole genome alignments, then there are smaller rearrangements going on. But of course, it's harder to quantify their effect on recombination, for example.
Thanks.
Really nice talk. So I liked the elegance of this idea that by looking at the way that a particular window will change on your first principal component, you can identify that as a place where there's possibly some sort of interesting structural element which is not mapping along with the phylogenetic tree. But of course, for that to work, your data set needs to have some pretty strong structure to begin with so that the position in PC1 is meaningful, meaning that you need to have different populations or different species. But I'm wondering if you had a situation where you had a data set of one relatively well-mixed population, but you still want to know, can you find inversions or some other structure in this population? Could you flip it around, and rather than using the position in PC1, but looking at the percentage of variance that PC1 explains, meaning that if your population is well-structured, PC1 is basically meaningless. But as soon as you hit an inversion, suddenly PC1 is much more important. I wonder if that would work.
Yeah, no, that's a good point. So the percentage of variance explained is also an important factor. So this you can see this for these inversions very clearly. The percentage of variance explained of PC1 is almost 0, and then it goes to a very high number in these inversion regions. So that's something that indeed with less phylogenetic structure you still expect this to play a role. I mean, I think even within a population you could actually see something similar if the inversions are older than the rest of the genome. If there was some balancing selection, for example, even if it's a well-mixed population, if there was balancing selection, you can get older haplotypes in this region, so you could still get a different phylogenic signal. And also I think the signal of selection, so if there is a sweep in this region in your population, it will still leave a distinct signal even if it's just a single species you're looking at. Mm-hmm. But the variance explained is also an important factor.
Thanks.
Well, I found very, very interesting the talk, especially because I keep these embunas in the aquariums. So the thing is that I have seen them hybridizing and then back crossing with one of the parental species, like some pseudotropheus. But the thing is, in that case, the hybrids are fertile and can back cross. But I found very interesting this mechanism that you were showing about these inversions. How long does it take, for these inversions to occur? Because they are not in the first generation of hybrids, or because once they occur, back crossing is no longer possible, or more difficult.
Yeah, so the inversions are actually much older than the species. So the inversion is something they already carry in their genome. But if one species is fixed for the inversion, then you could say you don't see it. It's like it's normal again, right? If you don't compare it to anything else. But if you have two species and they are fixed different for the inversion state, and you cross them, then suddenly it becomes heterozygous in the F1s. It's never heterozygous in the parental species because they are fixed, but then they are heterozygous, so then recombination is suppressed. It does not mean that back crossing is not possible, but it would mean that any kind of adaptation which is in the inversion is inherited as a whole block. So it's not broken up, so you don't get a mixture. So it would not, if for example, let's say the species difference, the color difference between the species or the shape is encoded in this inversion, you would not get recombinants, which then are some kind of intermediate shape, but you would always keep the pure either one or the other parental phenotype, basically.
Okay, understood.
Yeah, it's not mixing the phenotypes.
And then very interesting the point you said about the fitness of the hybrids. That I remember years ago at the Zoological Society in London, I saw a talk about a selection against hybrid males because they were, in the wild, they were getting more aggressions from the males of the two parental species. So this would be a way of a selection against hybrids. I don't know if you are working on that or it goes beyond your scope, or you know something about that?
One very interesting observation we made is we kind of cross hybrids as well in our facilities, and we haven't quantified it yet. So it's only a qualitative observation. Is that it looks like the hybrid males, they are not aggressive towards the parental males, but only amongst themselves. So apparently they're much less aggressive to the males of the parental species than to the males of their own species. So of course, if there is such an effect, this could have an important implication in reproductive isolation. Similarly, the question is, who do hybrid females prefer? Do they prefer that male or the other male or a hybrid male? And there's some work from Ole Seehausen on crossing Victoria cichlids on that, but it's an interesting point.
Okay, thank you.
Thanks, Hannes. Maybe we stop here. I have to say, two speakers were very good. We are doing very well with time, so I invite you all to go to the garden. We have a coffee break now for half an hour. Thanks. So our next speaker is Maves Jami. It's a pleasure to introduce her today. She's a postdoctoral researcher at the Swedish Research, funded by the Swedish Research Council. She has a bunch of affiliations, mostly the Swedish Agricultural University and a bunch more that I don't remember. She's broadly interested in diversity and evolution, specifically on protists. Really cool. She did a PhD at the Uppsala University with Fabian Burki. And since she was working on long-read metabarcoding, so I think she will talk also about that. And currently she's working on applying environmental sequencing approaches using long-read data to uncover novel protist diversity, very interesting, and also evolutionary relationships. Thank you.
Very much. Can you all hear me okay? Yes? Great. Okay, so thanks a lot, Iker and Ida, for the invitation, and thanks for the introduction. So as Iker said, I'm going to be giving a very broad overview of metabarcoding and metagenomics and how I use it in my research. So I've divided my talk into 3 parts. The first, I'm going to introduce metabarcoding, tell you what kind of questions we can answer with it, and 1 case study from my research. And then I'm going to delve into metagenomics. And then finally, if there's time, I'd like to spend a few minutes going over some resources that might be of interest to people in the audience. And before getting into metabarcoding, I'd like to introduce protists. And so here you see the eukaryotic tree of life. And of course, everyone here, we're all familiar with plants, animals, and fungi. And you may note that these are just a few branches on the eukaryotic tree, and most of eukaryotic diversity is, in fact, microbial. And all of these different microbes, eukaryotic microbes, they're together called protists. And they're not only incredibly diverse, they're very pretty to look at. And so in case you're not so familiar with them, I'm going to show you some pretty pictures so you can bond with them a little bit. So protists include diatoms, these photosynthetic algae that are encased in these very beautiful glass shells. They include green algae, which are closely related to land plants. They also include dinoflagellates. Some are heterotrophic, some are autotrophic. And these are the guys responsible for the bioluminescence you might see in the night sea. And you might also know them as the endosymbionts of corals. Protists also include ciliates. These are, some of them can be very large. They're very hungry, and they eat bacteria and other protists. They also include radiolaria. These are marine protists, again, with very intricate shells. And the last example I'm going to show you is vampirellids, which, as the name implies, are microbial vampires. So they will make holes in algae and then suck out all the good stuff. So protists, they're not only pretty, but they're also incredibly important on our ecosystems, playing roles in primary production, so making oxygen, in climate regulation, biogeochemical cycling, et cetera, et cetera. And the question is, how do we study this amazing invisible diversity of protists that is all around us? And one of the ways we can do that is through metabarcoding. So metabarcoding, if you're not familiar with it, is basically where you take an environmental sample, and this can be water, it could be soil, air, whatever you're interested in. You extract DNA, and then you amplify a specific marker, which is usually less than 500 base pairs. What marker you use really depends on which group you're interested in. So for animals, it tends to be the CO1 gene, ITS for fungi, TRNL and RBCL for plants. And since I'm interested in protists, I use the 18S ribosomal DNA gene. More specifically, we sequence one of the 2 hypervariable regions of the 18S gene. This is either the V4 or the V9 region. Right, so you amplify your fragment, and then usually you sequence it with Illumina. So you get loads and loads of reads. And then by comparing your sequences to reference databases, you can identify what's in your sample. So it's really like using barcodes to identify what item is what at a supermarket, but applied to environmental samples and very high throughput. And over the past decade, 15 years, there have been loads and loads of metabarcoding studies. So this map, this is already outdated because it's from 2020, but it shows you all of the different places where samples have been collected for environmental sequencing. So it's really quite an astonishing effort. And sorry, and this is from 1,500 articles. So what can we learn from all of this metabarcoding data? It can help answer really fundamental questions like, who is there in your environmental sample? And a really great example of this is the Tara Oceans project. So here they've collected marine water samples from around the world, and they did metabarcoding of the 18S-V9 marker to look at protists. They found 130,000 genetic types of protists, so that's more than 10 times the number of species that's been described. And you can ask really, really basic but important questions, like what is the most abundant group of protists in the oceans? What is the most diverse group of protists in the ocean? You can, of course, also compare different environmental samples to look at how and why communities vary across space and time. And I have this example from a paper published last year where they collected fungal spore samples from the air from basically around the world. And they could then compare tropical and temperate fungal communities and look at seasonal variation and see what are the main factors, the main environmental factors that really explain community composition. It can also help you look at past ecosystems. And I found this really cool paper where they took sediment core samples from a cave in Texas. And then using metabarcoding, they were looking for the presence and absence of different plant species over time. And what they found is that during this cold period, a lot of plant species vanished, but then the communities recovered after this cold wave. And then metabarcoding is also really good for looking at species interactions. And so here, this example that I'm showing you in this paper, they wanted to find out what do different species of bats eat. And so they collected bat fecal samples and did metabarcoding. And then they were able to look at the dietary preferences of different bat species and see what kind of insects do they like to eat. So metabarcoding can help us all of these kind of questions and many more. And the reason it's so popular is that it's really high throughput, it's easy, it's cheap, and you can catch most, but not all the taxa in a sample. But of course, there are some limitations as well. So first of all, this is based on a single genetic marker, so it's kind of limited information. There's also primer bias. In practice, no primer pair is ever universal, so you will always miss out some taxa. And then the relative abundances that you get are often unreliable because there's a PCR step, which can introduce biases. And then, of course, another limitation of metabarcoding is the read length, because oftentimes we're using Illumina sequencing. The marker that we use is less than 500 base pairs. And that means that with these short reads, you have very little phylogenetic signal. And then that makes it really difficult to answer ecological and evolutionary questions that require phylogenetic context, and is really difficult to place novel diversity accurately within the tree of life. And this is where long-read metabarcoding comes in. So in 2020, we introduced a protocol of long-read metabarcoding for eukaryotes. So here we use general eukaryotic primers to sequence not just a big chunk of the 18S gene, but a large fragment of the ribosomal DNA operon. So this fragment is around 4,500 base pairs, and it includes the ITS region as well as the 28S gene. And then these amplicons are sequenced on PacBio. And this method has several advantages. The most important one, or at least the one that I'm most excited about, is that with these longer reads, you have more phylogenetic signal, and you're able to infer phylogenies of your environmental diversity. And this, essentially, it opens up a whole host of questions that you can now address that you could not address before. And so, for instance, we used long-read metabarcoding to investigate habitat transitions across the eukaryotic tree of life. And why habitat transitions? Well, there are major evolutionary events. For example, if you think about the move of animals to land, and they can trigger adaptive radiations as well. And one of the most difficult habitat boundaries to cross is the salt barrier. So this is the boundary between saline marine environments and low salinity non-marine environments such as soils and freshwater. And this barrier is very difficult to cross because adapting to this really large shift in salinity is difficult. And so our question was, how frequently have eukaryotes, and I really mean protists, have crossed the salt barrier? And to find out, we started by collecting 21 samples from several different environments, such as soil, freshwater, the marine sunlit layer of the ocean, and then the dark aphotic zone. And it's not so many samples, but the aim here was really just to get a representative diversity from each of these different habitats. And then very briefly, we did long-read metabarcoding of these. We sequenced them on PacBio Sequel 2 on 4 smart chips. And we got roughly 11 million raw CCS or hi-fi reads, which we could then process and cluster into nearly 17,000 operational taxonomic units, or OTUs. So these are meant to represent sort of species level clusters, but yeah, with a grain of salt. And then we also taxonomically annotated all of these. So then with our OTUs, we could build multi-gene 18S plus 28S phylogenies. And this is what our phylogenies look like. So we've covered, or our phylogenies include all of the major eukaryotic lineages, such as, so here you have fungi, you have ciliates, radiolarians, and so on and so forth. And now, of course, for all of these OTUs, we know which habitat they come from, and we can map that and visualize habitat preferences across the eukaryotic tree. So in this outer circle here, yellow is freshwater, orange is soil, and then the light blue and dark blue are the different layers of the ocean. And what I want to point out here is that essentially the yellows and oranges, they're quite intermixed. So that means that freshwater and soil lineages, they're quite closely related to each other, as well as lineages living in different depths of the ocean. But what you can see is a phylogenetic distinction between the marine and non-marine lineages. And this confirms that, yes, the salt barrier is difficult to cross. Now, of course, our tree does not include all eukaryotic diversity. And so in an attempt to increase the cover diversity, we took advantage of the mass of available metabarcoding data. And what we did is we focused on the 18S V4 metabarcoding studies, 22 of them. We took all of their raw data, processed them, clustered them, and then built phylogenies with them, using the PacBio phylogenies as backbone constraints. And the very nice thing here is that by including this extra data, of course, you can then detect more transitions. And so I'm showing you an example here where, so this is a part of a diatom phylogeny, and everything in blue is marine, everything in orange is non-marine. And all of these marine taxa, they came from this short read metabarcoding data. Great. So now that we had these more comprehensive phylogenies, we could ask questions like, have different groups crossed the salt barrier with different frequencies? Are some groups more adept? And to answer this, we looked at the number of transitions per unit of branch length. So in this cartoon phylogeny, everything in blue is marine, everything in orange is non-marine. Here we see a single habitat transition event, or in other words, a low transition rate. Whereas in this phylogeny, we see multiple independent transitions, or in other words, a high transition rate. And so we could do this for different groups and plot their transition rates from low to high. And since this is not a dated phylogeny, I'm not going to show you the numbers because they're kind of meaningless at this point, but it's good for comparison. So now here I've plotted the data, and each different colored curve represents a different group. And what we see is, yes, transition rates vary very greatly across eukaryotes, with fungi having the highest transition rates, followed by cryptophytes and ochrophytes. But at this point, we don't know why certain groups, why fungi, for example, are so great at being able to cross the salt barrier. Next, we asked in which habitat did major eukaryotic groups arise? And we used ancestral state reconstruction to answer this question. And I'm showing you the results here. So this is a summarized eukaryotic tree. And at the tips are different phyla or major groups. And then the pie charts show you what is the ancestral habitat for that particular group. So for example, given the data and the model that we used, we saw that animals likely originated in marine habitats. Great, that's consistent with what we know about animals. But what I really want to point out is two of the largest assemblages called Amorphia and SAR. So Amorphia, this is the group that includes animals, fungi, amoebozoa, and some other relatives. Based on the data that we have, we inferred a more likely non-marine origin for this group, whereas for SAR, this is the group that includes ciliates, apicomplexa, radiolaria, et cetera, For this particular group, we inferred a more likely marine origin, meaning that two of the largest eukaryotic mega-assemblages likely arose in different habitats. Of course, I do think we should take this maybe a little bit with a grain of salt, because the deeper in time you go, the more difficult it is to be confident of your ancestral state reconstruction. But it does enable us to ask these sort of questions in the first place. So just to conclude the metabarcoding section, this is a method where you use a single genetic marker from environmental samples to generate sequencing data. It allows you to, it enables biodiversity assessments and can answer a lot of ecological and evolutionary questions. And I would think long-read metabarcoding in particular is very promising because it includes multiple markers that you can use. And it also has, with long reads, you have greater phylogenetic signal. And that allows you to answer a whole other set of questions. Great, so now we're going to move on to metagenomics. So metagenomics, you once again start with your environmental sample. And let's imagine in this case that we have 3 species in our environmental sample. So they're, and their genomes are shown in different colors. In metagenomics, instead of sequencing a single genetic marker, you do shotgun sequencing of all the DNA present in your sample. So this is called a metagenome. And then typically, these reads will then be assembled into contigs, and then based on GC content, sequence composition, and the coverage in different samples, these contigs will be binned into different, sort of put into different baskets or bins. And these are called metagenome assembled genomes, or MAGs for short. So what can metagenomics tell us? You can, of course, again, ask who is there. And one thing I'd really like to point out here is that metagenomics gives a more unbiased representation of community composition, because you don't have any primer bias, you don't have a PCR step that's biasing your relative abundances. But the caveat here is that metagenomics might not detect the most rare taxa in the community, and I think Lisa already mentioned that this morning. But of course, this will, how much you detect will depend on your sequencing depth. But most importantly, you're not only able to answer who is there, but what they are doing in the environment. And again, I'm going to show you an example from Tara Oceans. So here, this is you can see their sampling strategy over here. They sequenced metagenomes from all of these different samples. And I should note that they collected samples both from the epipelagic, so the sunlit layer, as well as the mesopelagic. As you might expect, they looked at different marker genes for specific processes like photosynthesis, sulfur metabolism, carbon fixation, et cetera. And as you would expect, they found differences in abundances of these marker genes at different ocean depths. So this is, these are some of the things you can do with metagenomics. And not only that, they also found a large number of novel genes. That's shown in red in these bar plots. And specifically, what they found is that the number of novel genes was really high for in habitats that haven't really been explored that much. So, the Southern Ocean, as well as, all samples taken from the deep ocean. Metagenomics has also been used to build catalogs of genomes, primarily for prokaryotes. So this paper is from 2020, so already a little bit outdated, but here they gathered more than 50,000 MAGs, representing nearly 13,000 novel candidate species-level OTUs for prokaryotes, which spanned all bacteria and archaea, and these novel OTUs and MAGs are depicted in green in this phylogeny. We can then use these MAGs and genomes to study how genomes change and evolve, and the scale is really, you can go from mutation or like SNPs to genome organization. And so here the schematic shows basically how you would use metagenomes to study the evolution of single nucleotide variants. So you map your metagenomic reads to reference genomes, and then by doing that, you can look at how SNPs would vary across space and through time. And of course, apart from all of these questions, they allow a deeper investigation into how communities vary across space and time and what past ecosystems look like. So metagenomics is high throughput, has no primer bias, no PCR bias, and it also allows you to look at functional diversity. The limitation is that it's a lot more expensive compared to metabarcoding, because as you can imagine, you really need to sequence quite deeply to get decent coverage of all of your community members. And another thing you may have noticed is that I mostly give examples of prokaryotes so far in this section. And this is because metagenomics is currently not as popular for eukaryotes, although it's certainly becoming more and more popular. And the reason for that is eukaryotes just generally have larger, more complex genomes, right? So yeah, so eukaryotes can have very large nuclear genomes. For dinoflagellates, that can be up to 270 gigas. They have a lot of non-coding, repetitive DNA. The genes can be fragmented because of introns, and the genes can be difficult to identify de novo. So all of this together means that you need to sequence really, really deeply to be able to assemble eukaryotic genomes, and even then, it's very challenging. But metagenomics can be a really great potential tool for looking at organellar genomes, such as mitochondria and plastids, because eukaryotic cells often contain multiple mitochondria or multiple plastids, so that means there's more DNA in the sample. And they also have very small genomes, so their genomes are typically smaller than bacterial genomes. So in principle, metagenomics should work great for organellar genomes. And this is something that we put to the test in this project that I'm going to present now on a new deep branching algal lineage that we discovered using metagenomics. So we're going to focus on plastid genomes. What are plastids? You might be more familiar with the more popular term chloroplasts. So these are the organelles that do photosynthesis, but also a whole bunch of other functions. So how many times have plastids evolved? If we map where we find plastids on the eukaryotic tree, you will see that you can find them in many different places. And all plastids, or nearly all plastids in the eukaryotic tree, have resulted from this primary endosymbiosis event. It's where a cyanobacterium was taken up by a eukaryote. And this primary endosymbiosis event took place we know it took place in the ancestor of Archaeplastida. So this is the group that includes land plants, but also green and red algae and glaucophytes. Now, plastids have also been acquired by secondary, tertiary, quaternary, and so on endosymbiosis. So this is where eukaryotic cells engulf other plastid-containing eukaryotic cells here. And this is how photosynthesis has mainly spread across the eukaryotic tree. So there's been 2 independent spreads of what we call green plastids, and then a number of, we basically find complex red plastids. So this is plastids of red algal origin in many different parts of the tree. And this is the bit that's quite highly debated. And I'm going to be focusing on complex red plastids here. And then, of course, there's also been more higher order endosymbioses. So, right. So I want to spend a couple of minutes just going over these complex red plastid lineages because they're going to come up a few times in the rest of the talk. So first up, we have cryptophytes. This is a group of predominantly freshwater algae. And one very interesting thing to note over here is that these guys contain 4 genomes inside a cell. So you have the nuclear genome, the mitochondrial genome, the plastid genome, but they also have something called a nucleomorph. So this is the remnant nucleus of the red algal endosymbiont that was eaten up by the ancestral cryptophyte. We also have haptophytes. These are tiny marine algae. A large clade of them is covered in tiny scales, and here you can see a haptophyte bloom from space. Right, so these are the 2 main groups I want you to remember, cryptophytes and haptophytes. And I also want you to remember that these 2 groups are in different places in the eukaryotic tree. There's 2 other groups that I will very briefly introduce. The first up is ochrophytes. This is a super diverse group. It contains everything from diatoms and golden algae to giant kelps. And the mysozoans. So this includes dinoflagellates and the apicomplexa. You might know this is the group that contains the malaria parasite. Yeah, so these are the 4 main red algal complex plastid lineages. And there's many hypotheses about how complex red plastids have spread. I'm just showing you 2 hypotheses here. You don't need to pay attention, but really the point is there's many hypotheses. And the main questions are, how many endosymbiotic events took place? Who were the donors? Who were the recipients? And answering these questions is really challenging because most algae are uncultured. And to resolve these questions, you need more genomes, more plastid genomes. And so what we did here is we decided to mine Tara Ocean's metagenomic data for plastid genomes. So I've mentioned this a few times already, so by now you might already be familiar with it. But Tara Oceans was this global expedition that collected water samples from around the world and sequenced them very deeply to generate metagenomic data sets. We then did co-assemblies of the metagenomes. We did 11 different ones, so each focusing on a different oceanic basin. And then from these co-assemblies, we pulled out 660 plastid mags that we manually curated. And these mags are roughly 63% complete on average. So our first step, once again, was to build a phylogeny with these 660 mags and 179 reference genomes that we manually selected. And this is the tree that we get. So the different colors here represent different groups. You have another ring around, which indicates whether a taxon is a mag or if it's a reference. And then you have a second ring that indicates size, but we're just going to ignore that for now. OK, so as expected, we didn't recover mags from groups that are predominantly freshwater or are multicellular, such as glaucophytes and red algae. But we retrieved a huge diversity of plastid genomes from ochrophytes as well as haptophytes. And something that really caught our eye was this tiny little group of 4 plastid mags that didn't fall in any of the other lineages. And interestingly, these mags corresponded well with DPL2. So this is an enigmatic plastid lineage that's only been known from 60nest data, but we didn't have any other data on it before. So what can we say about this new algal lineage? How big is it? Where does it live? And to find out, we mapped raw metagenomic reads against the plastid genomes. And we saw that overall, members of this new lineage are really small. So I'm showing you results from 2 mags, but it's the same for the rest. So on the x-axis is size in microns, from small to big. And what we saw is that our mags were predominantly present in the smaller size fractions, but not so much in the larger size fractions. So we think they're quite small, probably less than 5 microns. And so for making it easier to discuss the group, we informally named them leptophytes, for lepto to mean small. So I'm going to refer to them as leptophytes from here on. We also saw that leptophytes are overall globally distributed, but they're really rare. So on average, the mean coverage was less than 5 at any particular station that you looked at, with the exception of lepto1, which was present in high abundance, so like up to more than 100x coverage in 2 Arctic stations. So next we asked, what is the position of leptophytes in the plastid phylogeny? And to find out, we inferred a lot of phylogenies using all sorts of different models, which I will not go into right now. So I will just summarize the results. So some of the models that we used recovered leptophytes as sister to both haptophytes and cryptophytes with varying levels of support. whereas other models recovered leptophytes as being sister to haptophytes. So it's clear that leptophyte plastids are related to cryptophytes and haptophytes, but their exact position is ambiguous. And then in terms of gene content, we saw that leptophyte plastids contain all of the core plastid genes. We also looked specifically at genes that are missing in haptophytes. So haptophytes tend to miss like around 18 plastid genes in their genomes. We saw that leptophytes contain about half of them. And then we also looked specifically at genes that are present almost only in cryptophytes and red algae, the rhodophytes. And we saw that leptophytes did not have any of these genes, so the same as haptophytes. So to summarize, leptophyte plastid gene content is more similar to haptophytes. Now, one thing to keep in mind is that plastid genomes may not reflect the host or nuclear evolutionary history, right? Because there's this complex endosymbiotic events happening. And so there is this open question: where do leptophytes belong in the eukaryotic tree of life? And to answer this, we focused on, remember I mentioned that lepto1, one of the mags, was found in high abundance in a couple of Arctic stations. So we focused on those metagenomes. We did a co-assembly of those. And then we looked for mitochondrial mags that could correspond to leptophytes, or to lepto1 specifically. So from our mini co-assembly, we pulled out 34 mags, and then we inferred a tree with them. And happily, one of them was in a very distinct phylogenetic position. So it didn't fall in any of the other groups, and it was sister to haptophytes with maximal support. And what's more is that the abundance of this mitochondrial mag corresponded really, really well with the abundance of the lepto1 plastid mag across Tara Ocean metagenomes. And so that made us really confident that we had indeed recovered the corresponding lepto1 mitochondrial mag. And then based on this phylogeny, we can say, okay, since mitochondria tend to recapitulate nuclear phylogenies, we think leptophytes are sister to haptophytes in the nuclear tree. Or they could even be haptophytes themselves, but we would have to isolate the cells and do morphology to find that out. So, putting all of this together, we propose two scenarios of how complex red plastids have evolved. And I'm going to focus just on the haptophytes, leptophytes, and cryptophytes, just to keep things simple. So scenario number one. So this is where you start with, okay, cyanobacteria gets taken up by Archaeplastida. That's great. And then you go from red algae to cryptophytes, a plastid transfer here. And then from cryptophytes, the plastid is transferred to the ancestor of haptophytes and leptophytes. So that's scenario number one. And this is the more parsimonious scenario, and this would be, and this is based on the plastid phylogeny where leptophytes are sister to haptophytes. The second scenario, this is based on the plastid phylogeny where leptophytes are sister to both haptophytes and cryptophytes, so leptophytes in a deeper phylogenetic position in the plastid tree. So in this scenario, you have, cyanobacteria to Archaeplastida, and then from red algae to the ancestor of haptophytes and leptophytes, and then the plastid is transferred from haptophytes to cryptophytes. And this is less parsimonious for several reasons, but also it would involve multiple independent gene loss events in these two lineages. So to summarize the metagenomics section, so metagenomics sequences all of the DNA in your environmental sample. used for a lot of questions. And mostly it has been used for prokaryotes so far, but it's increasingly being used for eukaryotes. And I think using it for organellar genomes, like no one's done mitochondria yet, so it's a really promising way of finding novel lineages and really shedding light on deep eukaryotic evolution. Okay, so hopefully this talk has made you feel a little bit curious about how you can use existing metabarcoding and metagenomic data to study the distribution of your favorite organism. And there's a lot of resources out there. I'm just going to cover a couple of them, mostly protist-based. And the first one, so if you want to comb through 18S metabarcoding data, I really like using MetaPR2. And what's really neat is that it has compiled data sets from many different studies. It processed all of the raw data in the same standardized way. And now you can access all of this data in an interactive website. So just look up MetaPR2. And what you can do is you can select your clade of interest, or you can input your own query sequence, and then you can see where your sequence is detected. And what's really nice is that you can also download all of that sequence data along with the corresponding contextual environmental metadata. My second recommendation, if you want to look through Tara Ocean's metagenomic data, use Ocean Read Atlas, which also has an interactive website. So this is based on, I think the search is based on k-mers. So you basically input your sequence, and then you can, the output will show you where your sequence is found and how the presence or abundance of yours of that sequence corresponds or correlates with different environmental factors that you can choose to display and so on. And then the last one, this goes beyond metagenomic and metabarcoding data, but I wanted to mention it anyway. It's called Logan. And it's a very useful tool to search everything in NCBI's SRA, so Sequence Read Archive. So this includes not just metagenomic and metabarcoding data, but it will also include all transcriptomic and genomic data sets. And so all you have to do is go to Logan Search, and you hit Query the Planet, and then you can enter your sequence. It has to be a nucleotide sequence, and I think it's 1 KB max. And then, yeah, you can see where your favorite organism is found, or maybe your favorite protein, I don't know, protein coding gene. So you can see where your sequence is found, and then there's all sorts of tables and which tell you exactly which accession number you want, and then you can go chase them down, etc. So yeah, and with that, I want to finish by acknowledging all of the people that I've worked with on these projects. And I want to specifically mention Fabian Berkey, Tom Delmont, and Daniel Volo. And thanks to funding, and thank you for your attention.
Thanks, Mavesh.
Yeah. No? Do you want to use this one? Or no? Yeah.
Can you hear? Yes.
Yeah.
Thank you. Thanks, Mavesh. Really interesting talk. Are there questions for Mavesh?
Thank you for the talk. I have one quick question on the beginning of the talk, but later, if you have time, I would like to discuss things with
sure.
you. But when you were talking about metabarcoding and you were showing the tree of the marine versus terrestrial origin, is there like a paper published with the tree, or is?
Yes.
Can you tell me the name? I didn't get it.
Let's talk at lunch. But yeah, but the trees are all available as well. Yeah.
Thank you.
Sorry, we have a problem with the air conditioning. Gracias. More questions? We try to solve it now during the lunch break. questions for Maves? Let's think about protists. Air conditioning is not so interesting.
I think this is
So mitochondria in flowering plants do
Yes.
crazy stuff, and they are in bits. They're not circular. They're all over the
Right,
place.
yeah.
And you can use it in other land plants for phylogenetic reconstruction, but not for flowering plants. Do you account for these at all?
I mean,
Sorry for
but
being
not really, because I was focusing only on ocean stuff anyway. So
Because you have them as outgroups in the phylogeny. They are part of the phylogeny. They're so you have next. You already went through them. So you have green plants. So I'm wondering if those green plants include any angiosperms.
Oh, right. Sorry. So I should mention that this is a schematic tree based on nuclear, what our understanding is of the eukaryotic tree.
Okay.
Yeah. So it's not based on.
Thank you. Perfect.
Yeah.
That was it.
Not based on mitochondrial data. Yeah.
One last quick question before lunch. Are we hungry? Okay. Then let's thank Maves once more, and I remind you that you can go from here downstairs much faster than there. We are back at 2:30. Thanks. Hola. Hello. Let's start with the afternoon sessions. I know it's very interesting, but you can keep talking at 5. There will be more beers. So let's start our afternoon session with José Antonio Godoy. He's a distinguished researcher at the Estación Biológica de Doñana, another of the CSIC centers in Sevilla. He did his PhD and postdoctoral experience as a molecular biologist on plants. That's not what he will talk about today. Since he arrived at the Estación Biológica de Doñana, he has been involved in creating the Laboratory of Molecular Ecology and has worked in many studies integrating genetics, ecology, and conservation, and that's what he will talk about today. He worked on several of the most iconic species of Iberian vertebrates, such as the Iberian lynx, the osprey, I had to look what that is, quebrantahuesos. No? Well, a bird. Spanish imperial eagle and many other very characteristic stuff. He developed many molecular tools for monitoring and managing genetic diversity of Iberian lynx in collaboration with the administration. And today, I guess that's what he will talk about. Thanks for joining. The stage is yours.
Okay. Thanks, Seeker, for the organization for the invitation. It's been a fantastic symposium so far, and thank you all for attending despite the siesta time and a wonderful lunch that we had. So I hope I will keep you awake in the next 50 minutes. So I'll be talking about conservation genetics. That was my assignment for the symposium today, and mainly using the Iberian lynx as a model species to illustrate what can be done in conservation. Well, this was already introduced. I was already thinking that there is no need to introduce this to you, the magnitude and the transcendence of the biodiversity crisis, but I will just insist on that the solution to that must be science-based, and we have this discipline to address this biodiversity crisis. And I want to emphasize what the diversity crisis discipline is. So basically, it's mission-driven. So we have a target to address this crisis, right? With urgency, right? We need to do that soon, and we need to somehow translate that into actions. And that sometimes means acting without complete knowledge. So I know that as scientists, we are very led to increase our knowledge base and postpone any decision. But we must realize that in crisis disciplines like conservation biology, we need to provide solutions urgently, so sometimes with limited knowledge. And of course, the complexity of the task requires some different disciplines to contribute. And among them, it's genetics. And genetics has been a late addition to this discipline, and I would argue rather incomplete addition to the discipline. Why genetics is important for conservation is based on microevolutionary processes acting in small or declining populations that are basically genetic drift that will reduce genetic diversity, that will translate into reduced adaptive potential, and on the other hand, inbreeding occurring in small populations with reduce heterozygosity, increase homozygosity, also for the deleterious alleles that will become expressed as a reduced fitness. So basically, reduced fitness and reduced adaptive potential will contribute to maintaining populations in their path to extinction. So we need to address this. And this is what theory predicts, but it's already being observed empirically. This is a large meta-analysis of genetic diversity in hundreds of species. And the conclusion is clear. Genetic losses are occurring globally. The good news is that if we act, if conservation strategies are implemented, this can be stopped or even restored genetic diversity, but we need to act. So this is the paradigm around the importance of genetic and conservation diversity, okay? Nobody is saying that genetics is driving species to become endangered. We already know the usual suspects for that. These are the processes that make population become small, but once they are small, then these processes start acting in a feedback loop that a small population will lose diversity, increase in breeding, that will translate into lower adaptive potential, lower fitness, and that population will still become smaller maybe. And that process will go on even if the original causes of the decline are removed. So that's why it's important to address this independently of other factors that originally contributed to the decline. So that's the role of genetics in conservation. Here are a few tasks, right? Generally, is to conserve genetic diversity, but also as a side task to apply genetic tools to help conserving those species and populations. I will not get into detail on all of this, but for example, this I will not talk much, but it's important. Well, we already heard about how species originate, how speciation occurs as a gradual process. So in conservation, admitting and realizing, recognizing and acting accordingly to the existence of lineages evolving relatively independently within a species need to be addressed and need to be taken into account. If we want to preserve all the adaptive potential of the species, we can ignore parts of that. But I will be talking mostly on this other task, is assessing genetic risks. So we identify the risks are loss of genetic diversity, accumulation of inbreeding. So that's what we want to measure in populations that have gone through population decline. And in terms of tools, I mean, we can apply very efficient tools for monitoring population, not only the genetics of the population, but also the demography of the populations. And that's really helpful for some species. And as I said earlier, it's not only that something needs to be done, but you have to somehow suggest what needs to be done to restore genetic diversity. So that's where suggesting strategies for improving the genetics of the population. And you can do that at different levels, but we can collectively talk about genetic management. And there are also forensic applications, so basically identifying where illegal trade materials are coming from, or trying to identify illegal prosecution and to help combat these additional threats. Okay, in terms of methodology, most of them have been already introduced to you, but we have a range of approaches, methodological approaches, to get the data we need to do these tasks. And first, we have traditional markers. By traditional markers, we mean, for example, microsatellite markers that have been extensively used in the past and in the origin of this genetic conservation discipline, and still being used. And in the other extreme, we have whole genomes, right? We have resequencing whole genomes. Can argue that it's the best approach. We'll have all the information in the genome. But I would argue that you don't always need that level of information. So traditional markers still have a role to play in practical applications. And in the middle, well, there are other options as well. Well, with molecular markers, you can go big. Conceptually, it's not a breakthrough. I mean, it's just more molecular markers are better. And you have technologies to apply molecular markers in large quantities. But in the middle step between markers and whole genomes, you have these reduced representation strategies, right? To sequence maybe 1–2% of the genome randomly. That provides you typically tens of thousands of markers directly from whole genome sequences. So what to use is one of the questions. It depends what the goals are, or the needs are. So markers are pretty useful. You can get a pretty good picture of the levels of genetic diversity and how genetic variation is structured among population just by using markers. And probably you will want to use markers for monitoring, especially when based on non-invasive sampling where whole genome approaches are especially challenging. So, on the other hand, what the qualitatively different kind of information you get from whole genome sequencing is locus-based. So, processes like selection or introgression cannot be really addressed, and all kind of processes affecting functional variation. You really need these whole genome sequences. And this is our model species for conservation genomics. I think it's an excellent model. I will try to convince you of that. But basically, if you talk about endangered species, it's really endangered. I mean, the Iberian lynx was at the brink of extinction only 25 years ago, more or less. So yeah, that ticks one of the conditions for a model species in conservation genetics. But I think the more important one is that it's actively being conserved. And this is important because on one hand, it means that people in the field are capturing animals, recording information and data from those animals, and those samples can reach you for genetic characterization, so that makes things easier. But on the other hand, probably most importantly, the fact that the population is being monitored and managed makes possible to give recommendations that will translate into action eventually. If you can convince them that that's important enough. So yeah, you have the tools, you have the opportunity to influence conservation. That's my main point here. I'm not sure that being such a high-profile species is an advantage. Sometimes it's not that. On the contrary, you have to deal with a lot of attention from public media. And well, over the years, we have developed high-quality genomic resources. So I think those are the main factors why we are happy with the Iberian lynx and using it as a model for conservation genomics. So this is the recent history of the species. So this is how the range contracted during the last the second half of the 20th century, from the 60s to the 80s to the 2000. By the turn of the century, there were only 2 remnant populations, and not more than 100 individuals in total. That was when the species was classified as critical endangered by UCM. By that time, Iberian lynx was considered the most endangered species in the world. Okay. And a few years, well, we've been working for the last 20 years, as I said, first with molecular markers, but by year 2010 or 2012, we started promoting the sequencing of the Iberian lynx genome. And we finally got the funds to do that, and the Iberian lynx became one of the first species to have their endangered species to have the genome sequenced with a reference draft genome. And it was made basically in Spain, so it was also one of the species to be to have a genome developed in Spain. This is the quality of that draft. Probably not the best for current standards, but that was enough for what we wanted to do. So we developed these resources and with the collaboration of huge national, international collaboration. And we also wanted to first address how bad is the genetics of the aberrant links. Okay? We have reasons to suspect that it was bad, but when we got the data, it was really bad in the sense that the aberrant links was a species with the lowest genetic diversity in the world, at least for those species that have been with whole genome data, but not only. So species that have been traditionally considered genetically eroded, like the cheetah or others, had higher diversity than the aberrant links. And if you go broader, this is an early review of levels of genetic diversity across all species. I mean, there are plants, invertebrates, vertebrates there, in a logarithmic scale, a huge range, but the aberrant links is right there. I mean, it's the lowest of all those species. And remember that the genetic diversity is also distributed in 2 remnant populations that are genetically differentiated. And diversity is low in both populations, but especially in one of them is Doñana population, right? Keep that in mind. The other one, I refer to that as Andújar population or Sierra Morena population. So low genetic diversity, what about inbreeding? So you know these tools that when you have whole genome data, you can assess inbreeding using these bands of homozygosity. So it's very spectacular that you have here the Eurasian links that is not that depopulated genetically, so no signs of long runs of homozygosity. This is the Andújar, the more diverse random population of aberrant links. You can see some and there, some runs, but this is links from Doñana. So almost the whole chromosome is a stretch of homozygous sites. If you put numbers, the level of inbreeding in Doñana approaches what you will have in the offspring of a selfing, right? 0.5 almost. And the levels in Andújar were lower, reasonably lower. And you know that the length of the runs can give a hint on how old the inbreeding events producing those runs were. So long runs are recent, short runs are older. So you see that Andújar has some homozygosity in middle-sized runs, indicating older inbreeding events, while the larger runs in Doñana, well, occurs in Doñana, indicating more recent inbreeding. So yeah, that was intriguing. So how did it come, the aberrant links, to have such a low diversity? So, and we also took genomics to give a hint of past demography. So how was the demographic history of the species? It was reconstructed with different methods, different data. This is PSMC based on individual genomes. And you see that first we see here is the aberrant links long-term effective population size is around a few thousand, 2, maybe 3,000. But the other thing that we learn from this kind of analysis is that there have been several bottlenecks historically. In fact, we learned that the decline of the aberrant links started a few centuries ago, not only the last century, but probably 300-400 years ago. It's true that it intensified recently, about that time, but comes from a long history of small population size. For example, these are the 2 remnant populations, recent history. You see Andújar maintaining relatively large populations for the last 500 years until this bottleneck decline started. And these are current sizes, effective population sizes, while Doñana, you see, have been small and isolated for quite a long time. We thought it was only decades, but it's more like centuries, in fact. So that explains why probably the combination of these small long-term population sizes plus serial bottlenecks occurring in the last few centuries, that probably explains the low levels of genetic diversity. Still, we wanted to address if this low genetic diversity was, well, how much it was due to the recent bottleneck and how much it was due to long-term low effective population sizes. So as you already heard, museums give the opportunity to address this by looking, allowing to look into past patterns, access historical patterns. That's important in conservation, as was already mentioned by Moses, that allows you to draw baselines. So what's the healthy genetic level for the species? The requirement for that is that to sample population preceding the bottleneck, right? At least. You cannot always, in the case of the lynx, probably will not be able to do that because the decline has been started so long ago that even historical levels are, historical samples come from already bottlenecked populations. But we can use these data to somehow reconstruct the process. And that's what we did. And we got to sample historical specimens of the species. First option, or obvious option, is come to these institutions, like the museum in Madrid. They host a lot of Iberian lynx. The other large institution hosting Iberian lynx specimen is our collections at EBD, at Estación Biológica Doñana. So those are the two main natural history collections in Spain. But we very early found that it was not enough, mainly because collections are very focused on particular areas and particular time periods. So, for example, here in the museum, there's a good representations of the populations of Montes de Toledo around the 70s, if I recall correctly, while in Doñana it is since the 60s, 70s, and mostly from the Doñana population. So if we wanted to, that's not even enough. We wanted to have a good representation of the geographical coverage of the species, but also a long time, right? So we resorted to sampling smaller collections in Spain. Sometimes there's local museums, maybe. Sometimes even smaller collections in high schools, for example. But the great effort and the most rewarding and at the same time the most challenging and time-consuming effort was sampling particular collections. I mean, specimens in houses, in bars, in shops. So here you see Mireia, she did this excellent job with the historical diversity of Iberian lynx, sampling a specimen in a local shop, somewhere. I can't remember where exactly. But she traveled a lot, knocked on a lot of doors. Sometimes she was received and offered the specimens or to sample the specimens. Sometimes they refused even to open the door. You must realize that some of these persons were worried that they had legal troubles just for having these lynxes. But we learned a lot from that. One message is there is a lot of resources, historical resources in private collections, small private collections. So if you want to sample that, take your car and travel around. The other thing we learned is that it has a very nice geographical and temporal sampling, but only after sampling this. And what else? I was saying something. Okay. So by doing this effort, we were able to sample a range of period of time and also many of extinct past historical population. We also wanted to go earlier in time, and that's through ancient samples. I agree with the distinction of historical and ancient, so we tried to sample both in collaboration with many paleontologists and archaeologists. We were able to collect around 60 remains, 60 materials, dated from a few thousand to to 180,000 years old. And these are, and we tried to, generate whole genome sequences from that, but basically we were only able to reconstruct mitogenomes, but whole mitogenomes. 10 whole mitogenomes that were already distributed across the historical or ancient range. So with this, we first took a look at what were the patterns in these historical populations using these PCA or FCA approaches. Here you see in blue and green, these are the contemporary populations. So Andújar and Doñana, separated in this axis, indicating they are very highly differentiated genetically. And here in other colors, you have samples coming from any other population that has become extinct. And you see a lot of overlap there. So indicating that probably the historical populations was, in fact, a meta-population made of different fragments, as you saw in the map by them. But these fragments were already genetically differentiated, well, very little genetically differentiated when compared especially to the current genetic differentiation between these two populations. And what happened with historical samples coming from Doñana and Andújar, right? So those are these darker blue. So you have different patterns here. Doñana, historical samples from Doñana kind of overlap with current samples in Doñana, indicating that, well, even if we go back in time, the population was already differentiated from this meta-population. The oldest sample, in fact, is here, and that's from the 1800s. This is the single oldest population sample from the Doñana population, and you see it's within that cloud of. So yeah, we could track drift arising from the population, separating Doñana from this genetic pool. While in Andújar, you see that the pattern is similar, but historical samples from Andújar of only 20, 30, 50 years ago were mixed with this. So this differentiation of Andújar from this kind of palmitic pool has been more recent. So for us, and as you will see in the few examples later, Andújar is kind of a non-bottleneck or less drifted populations that we can compare to the highly drifted Doñana populations. Okay, so that we could reconstruct the history of each of these fragments genetically and demographically, and you see this clear contrast in demographic history between Andújar, that was was large and connected only a few decades ago, while Doñana separated from this palmitic population centuries ago and has remained small since then. Okay, so this is kind of what we inferred, kind of relatively diverse, I will go back to this now, palmitic population in the ancient past, historically a meta-population with different levels of genetic differentiation, but not that high, and currently two isolated and highly inbred populations, one in Doñana and Andújar. And we could also estimate what's the diversity when compared to this historical diversity that remains in each of these two populations. So Doñana has lost about 50% of the historical diversity, Andújar only 84%. But the good news is that if you take both populations together, then you kind of recover heterozygosity, about 90% of the heterozygosity present in this meta-population. So what happened here? These are nuclear, these are mitochondrial patterns. We couldn't get nuclear data from those ancient samples at that point. And this is based on microsatellite data, I must say. These are mitogenomes, these are microsatellite data. But later, we wanted to insist. So we had these samples that were good enough to provide mitogenome data. So we went back to those samples and a few more that look promising, and redo the extraction, the library construction, and try to get nuclear whole genome data. And we managed to, with only 3 samples, one from here, one from Catalonia, and one from Andalusia, that range between 2,000 and 4,000 years old. And for those, we got some whole genome resequencing at a depth of between 2 and 4x. So the question is, okay, our expectation, probably your expectation, is also that ancient sample will have larger diversity, higher diversity than historical or modern. However, we found that ancient samples have lower diversity than contemporary samples. Yeah, that's struck us, really. So the diversity in the ancient sample was kind of similar to the one in Doñana currently, but only half of that present in Andújar population nowadays. So that was striking. I mean, we needed to explain where that diversity came from. So, and the answer came when we analyzed introgression. So this is ABABABA test, the D statistics, and you already heard about that. So the more extreme that is, the value indicates introgression from these 2 species. So we detected, when we compare ancient to contemporary Iberian lynx with Eurasian lynx, a sister species, we find that contemporary lynxes have a lot more introgression than ancient lynxes. So that could certainly explain why current samples have more genetic diversity. It came through introgression with Iberian lynx. So that could suggest that introgression has been either recurrent or continuous during the last 4,000 years, and that has impacted genetic diversity. It was not a complete surprise, because we knew from more phylogenomic studies that Eurasian and Iberian lynx have hybridized extensively in the past. So there are signatures in the genome that this hybridization has been extensive between Iberian and Eurasian, to the point that the majority, that the topology of the tree relating the 4 lynx species in the genus has been misled by this introgression. So if you wonder, the consensus topology would be one where Pardinus is the Iberian lynx, is sister species to the Eurasian lynx. But if you focus on introgression-free low recombination regions of the genome, or the X chromosome, large part of the X chromosome, then the topology you reconstruct is an ancient divergence, older divergence of Pardinus, and the extensive introgression between Eurasian and Iberian lynx. Okay. At what time am I supposed to finish? Only 5? Okay. Okay, I need to run. Okay, this is what I already showed you, but I was kind of, that was a simplified version. So now with whole genome data, we want to really understand not what happened with neutral diversity, but with functional diversity, and for that, we need to complicate this with selection. I mean, selection will act on functional diversity and will usually reduce diversity, but drift can interfere with that and make moderately deleterious alleles to accumulate rather than being eliminated from the population. And so that genetic load, the amount of deleterious alleles, increase in time in these populations, and eventually part of that load could become purged, eliminated, because of inbreeding expressing these deleterious alleles. So we wanted to address this by comparing what happened with the relatively non-bottleneck and bottleneck population. In the Iberian lynx we have 2. We selected another 3 populations in Eurasian lynx, and we directly compare how genetic diversity is reduced in the bottleneck with respect to the non-bottleneck. And we have different levels indicating different intensities of those bottlenecks from 50 in the Iberian, 70 or 80 in the Eurasian. And we wonder, okay, how, when we compare diversity in the bottleneck to the non-bottleneck and make a relative difference, how different features in the genome compare. And what we found is that most selectively constrained features like coding or even non-coding ultra-conserved elements will have increased diversity in the bottleneck population, indicating relaxation of purifying selection, probably increasing load. But at the same time, when we look at genetic load more explicitly by annotating the functional impact of coding variants, and we measure the load in individual genomes, so the counts of derived alleles in different functional categories, intergenic, neutral, and missense tolerated or missense deleterious, and we compare these same 5 populations, we see that deleterious derived alleles are equally common in neutral features, also in missense tolerated features, but that amount is lower in Iberian links with respect to the Eurasian links, indicating some level of purging also. That means that probably the consequences of further inbreeding in the population will be lower in the Iberian links than in the Eurasian link, but not that inbreeding does not have an effect. And we have several evidences suggesting that inbreeding adaptation does occur in the species. Probably the more direct is those indicating that more inbreeding leads to higher proportion of abnormal alleles. So we may wonder if the Iberian lynx is within this extinction vortex. And we can say that, yes, in fact, we showed that Iberian lynx lost diversity, accumulated inbreeding that affected functional population, that increased genetic load. We have some evidence that this is affecting fitness, so it seems like there is some reason to believe that this is happening. And we need to provide solutions, right? We need to suggest what to do now. And the solution for inbreeding depression is gene flow. Okay, so the obvious action to take is, well, let's mix those two populations. We've been lucky enough that the two populations persisted, because if only one population persisted, we will be unable to do anything. And the only concern is, should we mix them? Will be any detrimental effects by mixing those two populations? And the easy answer is no. Same species, no evidence for local adaptations. They have been connected in the past, maybe a few hundred years ago. So no reason to anticipate the deleterious effect of mixing. So the species have been mixed different ways. So this is a cartoon showing what conservation of the Iberian lynx is currently looking. I mean, it's how it's organized. We have the two renan populations. Animals from the two renan populations were brought to captivity, and there is a program that has been really successful in producing calves and maintaining genetic diversity within captivity. And in the latest years, animals born in captivity have been released into reintroduction areas. And on the other hand, remember that Doñana was the best candidate to suffer inbreeding depression and the more in need for action. So translocation have also happened between moving individuals from Andújar to Doñana. That has also helped the population. So all this has been managed genetically using, again, markers, but markers that have been selected from whole genome data. So they are optimal markers to do the job in terms of information content and power to discriminate individuals, assign parentage, or estimate kinship. And there has been a lot of huge effort to sample these populations non-invasively using feces. We have genotyped more than 5,000 feces in the process. And that has allowed us to orient the genetic management of these actions. I will go through this. This is, for example, the effect of translocations in the Doñana population. So this is where the first animal was translocated. This is diversity being reduced over time in the population. This comes from back in time. And soon after the translocation, the diversity increased. This is representing heterozygosity, and especially F1 hybrids with high heterozygosity here in the first few years. And this has been, the diversity has increased. This has occurred only because of high success of one single individual. So this is creating now problems of inbreeding coming from the excessive success of this individual and their descendants. But the net effect, so the big question is did this help the Doñana population? And if you remember, this reconstruction of demographic history of Doñana shows that in the last several decades, Doñana population had remained small, 50. Hasn't been any growth despite huge efforts in conservation. So to the point that in the latest years, Doñana animals have been fed, basically, provided illimited resources in terms of prey and feed. And despite that, it was not growing. But soon after the translocation, the population started to grow. So I think it's a good example of what can be done with populations that has such a high level of erosion and what we call genetic rescue. So, okay, I will skip this just to show the current status of the species. We have gone from 100 individuals to populations just in the early 2000s, to more than 2,400 individuals in the latest census. That's a huge success of conservation effort, and one where genetics has been probably part of the problem, contributing to the decline at one point, but also part of the solution when you just manage the population genetically. So that has led to from critically endangered, last year it was reduced to vulnerable. Okay, so do I have 2 minutes more? Good. So just a reflection. Okay, so what can we expect from population genomics from now on? Okay, I hope I made the point clear that conservation genomics need to provide solutions, right? So I appreciate all the effort in generating reference genomes. I think those are huge resources, very valuable resources, but we need to go beyond that to be able to impact conservation in practice. But in terms of generating new knowledge, there are a few aspects where conservation genomics can expand, certainly addressing, including other sources of variation. Structural variation should be especially relevant for fitness, and we have been missing that largely. Epigenomics, but again, what would be a qualitatively different contribution to conservation was the ability to improve fitness through the management of the really important genetic variation, either by reducing genetic load, maybe managing deleterious traits in the populations, and we're not still there. And we have huge, we are trying to go into that direction with an ongoing project, generating these missing layers of variation. And as I said, the good advantage of Iberian lynx as a model is that you also have fitness data. And all the monitoring performed in the reintroduced populations is providing survival, reproduction data that we can cross with the genomic data to be able to address the genetic basis of inbreeding depression and to follow this rescue effect after the admixture of the two populations with the prospect of ambition of identifying those losses that were responsible of depression, but also of improved fitness. But the large challenge of conservation genomics is this, translating that into practice. I mean, it's not always in our hand as a genomic and geneticist. So a lot of work needs to be done in closing this gap. And that is being done, in fact, by different initiatives. So, for example, the IUCN founded the Conservation Specialist Conservation Genetic Specialist Group only in 2014. And there is a lot of effort trying to bring managers and law-making persons, policymakers, to the community to make them realize the importance of genetic diversity and transfer them the tools to address that. So I think that's it. I thank you to my group, and thank you for the funding and collaborators, and thank you all for attending. Yeah. ¿Ahí hay otro?
Hola. Sí, that works. Thank you, José Antonio, for the nice talk. I appreciate that it's a talk about conservation genomics with a happy ending. That doesn't happen too often, so that's really nice. Do you have some quick question for José Antonio? With these beautiful images of the Iberian lynx.
Hi, I have maybe a naive question. So you talked about this population increase in the Doñana subpopulation after this translocation, and I'm wondering, what is it specifically that changed? Was it more pups being born, or I don't know, higher survival of the pups, or what was it that actually made the populations increase?
Yeah, we need to formally address that. We are taking a look to that. I mean, I think that the missing link we have now is just showing that better, improved genetic diversity is the cause of the increase in demography, in numbers. And we have some evidence that, for example, clearly at mixed individuals, especially F1, has increased reproductive success. I think probably both, because we documented smaller litter size in Doñana over time, and that has been restored now after the reintroduction, the translocations, but also survival. I mean, one episode in Doñana, a virus, it was feline leukemia virus, entered the population, killed a large proportion of the males in the populations 1 year. That was dramatic. So indicating that probably response to infectious diseases was limited in the population. That we haven't reintroduced the virus now to do the experiment. I think they will not allow us to do that. But hopefully, one of the things that have been restored is the capacity to respond to this. We know that an allele missing, an MHC allele missing in the original population has been now introduced with the translocation. So.
I was wondering, you just mentioned kind of defense against pathogens, but it could also go the other way around, that you actually introduce new pathogens when you translocate individuals. You haven't really mentioned that. Is this something that you kind of also very careful with? Of course, you can. I mean, there are some pathogens you might not detect when you just have them in captivity, right?
Yeah. Yeah, translocated animals are put into quarantine, but there's always a risk of introducing new pathogens in the populations. So far, it hasn't happened, or we're not aware of that happening.
One last question.
You kind of showed already that the population decline started like 400 years ago.
Sorry?
You showed in one of your graphs that the population decline already started like hundreds of years ago, right?
Yes.
Do you know, is this linked with human expansion into the... Is there any known source for this?
Yeah, we have
hunting
speculated a lot on that. It's about the discovery age, where a lot of traveling around the world with ships and that. But again, Iberian lynx is not a forest species necessarily, so that. One thing is the two ideas we have. One is powder, the guns, and we know that historical records show a huge, very expensive price for lynx coats during that period. So maybe that was persecution directly but we don't know, really don't know.
Okay, I think with this we can go to the next talk. Thank you very much.
Most of you remained awake.
So, and like this, it was all very quick, but we arrived to the last talk of the day, which is by Eric Dexter. He's a research scientist with expertise in genomics, molecular biology, bioinformatics, and data science. His research interests are around developing statistical models to investigate spread of invasive species, genetic basis of bacterial infection, and the structure of animal genomes, among other topics. He completed his PhD at Washington State University, followed by a postdoc in the University of Basel, working with Dieter Ebert. And he has even established his own consulting company, specialized in data science and bioinformatics. And today he will talk about how we can apply genomics to study species interactions. Thank you very much.
Okay, so the first question is, can everybody hear me? Are we good? All right, good. So thanks for being here. Thanks for sticking through to the very end. I will try to keep it fun and entertaining for everybody. So let's ask the basic question of why do some animals, plants, bacteria, viruses, why do they get sick when others don't? So let's think about that from a genomics perspective. But first, I want to get to some definitions that I'm going to be using a lot throughout this talk, so let's get it out of the way. So I'm going to talk about coevolution, and by that I mean something very specific, which is when you have reciprocal evolutionary changes between two very closely interacting species. This can be a host and a parasite, it could be a pollinator and the plant that it pollinates, but it's a situation in which an evolutionary change in one of the partners in this interaction forces an evolutionary change in the other one, and so forth. I'm going to talk about parasites a lot, and by that I mean it very broadly to encompass bacteria, viruses, animals, protozoans, anything that's living in a very close association and causing some harm to their hosts. That's the lens through which I'm going to study coevolution, but of course, coevolution can also be a mutualistic interaction. I'm going to be talking a lot about the specific statistical method of co-GWAS, a co-genome-wide association study. That's the framework that I'm going to use to look for the signal of coevolution. And then finally, I'm going to be talking about interlinkage, which is a new term that I'm trying to promote, which is interspecies linkage disequilibrium, which is this idea that when we have these very tightly involved interactions, we would expect that we could find a statistical correlation between alleles and genomes which are belonging to different species. So linkage disequilibrium across genomes. This is what I'm really interested in finding because this is a predicted genomic signature of coevolution. And I want to emphasize predicted because I'll let you decide by the end of this talk, but this is something which has never been directly observed before. So let's begin with a very basic model and think about, when I say the statistical signal of coevolution, how do we put some numbers behind that? What would that look like? If we can imagine a hypothetical system here, it's incredibly simple. We've got a host and we've got a parasite. And the parasite's ability to infect that host is entirely genetic based. But we don't have a universally infective allele or universally resistant allele in the host. It's context dependent. So we would say in this case, if the parasite has big B allele, it can infect hosts that have the big A allele, but not vice versa. And then conversely, we have the opposite here. So that there's no sort of universal winner or loser. The fitness of any of these alleles is context dependent. And thinking about biodiversity, we can see how this type of system, if you approach it from a sort of a game theory perspective, is something that's going to reward diversity. If we think about balancing selection, if you're in a population where most of the hosts have the big A allele, there's going to be very strong selective pressure favoring hosts which have the allele which allows them to infect that. But of course, then you're going to have a strong benefit towards having the host little a allele, and suddenly now the fitness landscape has flipped. And so you have these cycling dynamics in which simply having a rare genotype is selectively advantageous, and having the common genotype is a disadvantage. We can also see this would promote diversity of not just alleles at one gene, but diversity of copies of the genes. Because here, the parasite has to choose. It can have the big B allele, or it can have the little b allele. If you could duplicate the infectivity gene, suddenly it can express both at the same time and infect everything. Additionally, if you have an individual which comes in which carries a new allele, say the C allele, which can now infect both of the host alleles, suddenly the host has to respond with an increased, corresponding increase in diversity. Because once you have a universal winner or a universal loser in the system, it stops functioning. Now, this seems very, very simplistic, but this is actually a really great framework to ask questions about coevolution. Because we can say that if we go out in the wild and we sample these hosts, when we find infected individuals, we can only find specific genotype combinations because only some combinations result in an infected individual. So we can map this onto a field study now. Now, the system that we're going to use to explore this concept actually works very, very well. This is what I've been working on in Switzerland. We have our host, which is Daphnia magna. It's a planktonic crustacean. It's very small, about the size of a lentil. And you can find them in fresh water basically throughout the entire northern hemisphere and in some populations in the southern hemisphere. They tend to have huge population sizes, and they're just filter feeding in the plankton. In the wild, Daphnia magna is naturally infected by a bacterial parasite called Pasteurella ramosa. It's a spore. It's just picked up passively through the filter feeding process. When it encounters a host to which it can infect, it attaches to the gut lining, proliferates in the body cavity, and fills the entire body cavity with spores. That's what you see here on the top. We have this individual. It's dark because its body cavity is filled completely with Pasteurella ramosa spores. Eventually, these will break open, or that the body cavity will break open when the Daphnia dies, floods the environment, and propagation continues. Or nothing happens, which is what you see on the bottom right there. You have a healthy adult female. That's because whether or not an individual strain of Pasteurella ramosa can infect a particular Daphnia genotype is 100% genetically determined. It depends solely having a matching combination of infectivity and resistance genes on both sides. The condition of the Daphnia plays no effect, and mortality is 100% if it's infected. So very high selective pressure and binary outcomes. A particular combination of host and parasite genotypes will either A, always result in infection, or B, never result in infection. And from that, based off of previous laboratory work, we've been able to reconstruct actually a type of matching allele matrix. This is based off of real data with the Daphnia magna Pasteurella ramosa system. We can see now, instead of dealing with alleles in the top, we're dealing with two different isolates. These are lab cultured isolates of Pasteuria ramosa. They're just called C1 and C19. And then when we have hosts, we're looking at two different known resistance alleles. They're all in Daphnia, all of the resistance genes have letters. So if I talk about the A locus, the B locus, the C locus, et cetera, these are all different Pasteuria resistance genes in the host. There are epistatic interactions, but we know for this particular panel of C1 and C19, if we know the allelic state in the host at the A locus or the B locus, we basically have perfect predictive ability to determine whether or not an infection will result. So this matching allele model does map very well onto the system. It's just that the matrices are more complicated. So this seems like a great system to look for the signal of coevolution. We also have a fantastic field site. This is Lake Egelsee, which is not so far from Basel, about an hour away. And for, 10, getting close to 15 years now, the Ebert group has been going out and sampling this lake regularly because there's a fantastic population of Daphnia magna here. They overwinter during the winter months, and in the spring, they emerge from resting stages, and you will have basically Daphnia soup in this lake, billions and billions of individuals. So very large population, very well mixed, but also very discrete. And we also have Pasteuria in this population here. Here's a data set looking at just field sampling from just 5 years of what is now a much larger data set. And what we have on the y-axis is the prevalence of Pasteuria infection. So we go out throughout the course of the season, we collect random Daphnia, bring them back to the laboratory, determine whether they are infected or not. And we find that every year there's this massive seasonal summer spike of Pasteuria infection, where upwards of 99% of all of the Daphnia in the water column are infected by Pasteuria ramosa. Now, they will all die. None of them will recover from the infection. The only way out of this is to die from something else, which means that by the end of the season, the pond is comprised entirely of individuals which have somehow escaped Pasteuria infection. And we see not only this change in abundance, but if we now take individuals from the same sampling periods and we phenotype them in the laboratory, because we can raise the Pasteuria and Daphne in the laboratory. And we do an experimental infection assay where we take the Pasteuria isolate, which we call P20. This was isolated from this field site, and it's the most common Pasteuria strain in this field site. And at that point in time, we take large cohorts of Daphnia, bring them back to the laboratory, and we try to infect them with P20. We see at the beginning of every season, about 50% of the individuals are susceptible to P20, and about 50% are resistant. But we see by the end of the season, the trait of P20 susceptibility has basically disappeared, which makes sense because they went through this massive selective event. And then for reasons that we're not going to talk about today, at the beginning of every spring, P20 resistant or susceptibility reemerges in the spring. That's a very interesting dynamic. Why doesn't it just go away permanently? But don't have time to talk about that today. But the important thing is we see that every year there is this very, very strong selective event always pushing in the same direction. So it seems like we have everything in place now with this system. If we're going to find this signal of interlinkage anywhere, it seems like this is a great place to go looking for it. So let's talk about the experimental design for how I did this. So the first off is going back to this field site, and throughout the course of one field season, every few weeks, I'm just collecting cohorts of wild Daphnia, typically several hundred individuals at a time. And I bring them back into the laboratory. One of the great things about working with Daphnia as a model organism, they will naturally reproduce asexually. So you can create clone lines in the laboratory, but you can also cross them. So you get both asexual and sexual recombination. So from those wild Daphnia, from each one of the individuals I collected, I immediately start a clone line, and we keep that clone line going. And then I monitor the mother to see if it was infected with Pasteuria ramosa. If it was, I then sequence the mother. Whole genome sequencing, the entire animal, the whole Daphnia goes right into the tube. And we're sequencing not just the Daphnia, but we're sequencing the genome of the Pasteuria within the Daphnia. Of course, we've lost the mother, but we still have the genotype because now we have the clone line going in the lab, and we can do experiments with that. We bioinformatically separate the host genome from the parasite genome. So we get 2 whole genomes from one sample. And then with the clone line, we then do a bunch of infection assays to determine what is the resistance phenotype. From our large panel of Pasteurial isolates in the laboratory, which ones can infect that genotype, which ones can't. On the DNA side, the host genome and the parasite genome goes into our co-GWAS statistical model. I'll talk about that a little bit more in a bit, which gives us some really nice statistical results, which of course could really end up being a fishing expedition. So we want to have some sort of biological validation that what we're finding with our co-GWAS actually has some biological weight behind it, and not just simply a really enticing set of p-values. Now this all centers around the co-GWAS, so I want to take some time and really explain what this is. I think we're probably here all familiar with the concept of a genome-wide association study. Here's just some example pulled from the literature looking at human data, and we're looking at sort of the typical way you represent a GWAS result. We have our Manhattan plot, and we're looking at, we've got 22 autosomal chromosomes, and this particular GWAS, there was a particular human disease phenotype, and they wanted to try to map and figure out which genes in the human genome are maybe responsible for that. You get these really nice peaks, that's why they call it a Manhattan plot, sort of like skyscrapers. You assign some sort of threshold for statistical significance and say, okay, these are my candidate locations where I think there's a genetic contribution to whatever trait it was I was looking at. So for a co-GWAS, I'm going to do that. But I'm not going to do it just one time. I'm going to do one GWAS for every single allelic variant in the parasite genome and ask position by position, in the Daphnia side of the genome, is there a statistical correlation between having this allelic site and having been infected by a bacterial strain which has a particular allelic site? You can sort of think of that as doing a series of 10,000 GWAS Manhattan plots, stacking them all up on top of each other, and we're just going to look down through the stack and find where are the peaks. Something that I've chosen to do with this study is assigning significance thresholds. I'm not going to do it. I'm not going to deal with multiple corrections. I'm going to say our p-values are simply helping us identify the strongest candidates, but I'm not going to assess statistical significance at any level. The co-GWAS model structure, I'll just talk about briefly a little bit. Basically, it's a massive series of linear regressions. The most important detail is we're basically doing every pairwise comparison for every possible host SNP or short indel versus every possible parasite SNP or short indel, which gives us 14 billion linear regressions. You can see this now becomes a computer science problem because we have to numerically optimize how do we run this. This is brute force at this point. If I just set out and run this on a local machine, this is 3 months of processing time. And that's assuming I set up the model right the first time and don't have to rerun it. So there was a long period of time of numerically optimizing this, having to do a lot of matrix multiplication to try to get this faster. And I got the model down from an initial run time of 30 days down to about 30 minutes, which was really important if this was something I was going to actually experiment and iterate with. Okay, so we're getting a statistical model. I said we're going to have p-values at the end. We're going to use those as candidates. But at the end, I really don't want to be doing a fishing expedition, and I don't want to go interpret a model which might be sending me down a blind alley. So I want to know that my model can produce valid results. Fortunately, on both the host and the parasite side, we already have a pretty good idea of at least what some of the genes involved in interaction are. So if my model can't pull the known genes out in the system, I'm not going to trust it to send me out to find these new mysterious unknown genes. On the Pasteurial side of the system, we have a series of collagen-like proteins, PCLs, Pasteurial collagen-like proteins they're called. And we have pretty strong evidence that these are the mechanism of infection, or at least this is responsible for the binary outcome. Collagen-like proteins are more typically associated with the animal kingdom, but bacteria do have the ability to produce them. But what's unusual about Pasteuria is that it produces, well, we have I should back up a little bit. Why do bacteria produce collagen-like proteins? One of the main purposes is they are a mechanism of attaching to the host. So it tends to be a feature of pathogenic bacteria, and it is the mechanism by which the bacteria knows it has just encountered its host. Basically a lock and key mechanism where the collagen protein binds to the appropriate host tissue. Bacteria species which do have this mechanism tend to have between 1, 2, or 3 copies of the collagen-like proteins. Pasteuria ramosa is an anomaly, and that we found that it has between 40 and 50 copies of these collagen-like genes, and it varies tremendously between individual isolates, not only in terms of the copy number, but also the location of them. We've also found that these collagen-like genes often exist in these little triplet formations where we have 3 subfamilies, and we find a cluster where we have a member of each one of the subfamilies connected together. And these clusters are highly mobile between isolates. It seems like transposable elements are in some way moving these together as a cassette. But we also have many collagen-like proteins, which are singletons, which are duplicates. We're not quite sure what's going on there, but the hypothesis is that this triplicate structure is functionally significant. So we hope that our GWAS model is able to find the ones we know about in order to have some confidence in it. On the host side, we also have a pretty good idea of at least what some of the resistance genes involved are. I said they all have letter names. We have the ABC locus, called so because these are 3 loci which function basically like a supergene and co-segregate. We also have the D locus, the E locus, the F locus. These will all epistatically interact with each other to produce the resistance phenotype. So we've got a pretty good idea of what we hope for our model to recover. So let's get into some of the model results now. So now this is our Manhattan plot. We're looking at just the peaks. Now we're looking at the Daphnia magna genome. So again, this is actually 10,000 GWAS plots all stacked down, and we're looking down the stack. And so these are the peaks that emerge out of all of them. We've got 6 peaks. And the question is, okay, so what's at these peaks? Well, the biggest one is the ABC/the F locus. They sit right next to each other, so we can't differentiate them. Okay, that's great. That makes sense. Next, we recover the D locus. Next, we recover the E locus. So in an entirely candidate-free way, our statistical model has just discovered the resistance genes in the Daphnia genome. Really fantastic. So what are these other peaks now? Do we have exciting new discoveries? These, I think, are mapping errors. These are homologs to the Pasteuria resistance genes and other loci, but they appear to be pseudogenes. So I don't think anything's really happening there. And that we don't know. This is a great question for a graduate student to investigate what's happening at that peak. But given that our model seems to be performing pretty well, I think it's worth looking into. So I said I'm interested in coevolution, but we're only looking at one genome right now. And all I've done is find things that we already knew. So that's great, but we didn't learn anything yet. The real question we have is, we've got this large set of resistance genes in one genome, and this large set of infectivity genes in our parasite genome. The question is, how are they interacting? And we can get to that really nicely now that we have some confidence in our model. So we're going to look here at this circular plot where I'm looking at, this is just nucleotide diversity, just pi. We've got our host genome on the bottom side of the plot here. We have 10 chromosomes. And on the top, we've got our Pasteuria genome. It's an order of magnitude smaller than the host, but I've sort of scaled them to the same so we can see what's going on. In terms of nucleotide diversity, there are some interesting peaks on the bottom of the host, nothing I would interpret too much. But on the Pasteuria side, we see, indeed, we do have some pretty big peaks of nucleotide diversity. That's interesting. But now what happens when we stick our Manhattan plot results as an inner ring inside of here? On the bottom, we have our GWAS Manhattan plot that we just saw before on the other slide. The only difference is it's upside down and stretched, but we still have those same 6 peaks. But now we see we have a corresponding Manhattan plot on the Pasteuria side. Also some really nice discrete peaks there. Here's the really exciting part, is we figure, okay, what are at those locations and how are they connected together? We see here, we have our Pasteur resistance loci, each being connected to discrete regions of the Pasteur genome. And if I annotate them a little further, the red lines here indicate the location of the collagen-like genes that are located in this triplet formation. So right here in one hat plot, we have basically an ideal confirmation of our entire understanding of how this model works. We see that the hypothesized resistance genes have an extraordinarily strong statistical correlation with the hypothesized infectivity genes. We see the specific pattern of genes in which epistasis that we see in the laboratory now makes sense because we see, for example, the C locus and the D locus are both interacting with this set of triplets on the sort of top right there, which sort of is another way of visualizing the epistasis we see. And we see that this triplet conformation of the collagen-like proteins apparently is very functionally significant because all of our interlinkage is always between triplets. It's never between the single collagen or the duplicate collagen genes. So the conformation appears to be important for functionality. So I would say here, we have at this point very clear evidence for interspecies linkage disequilibrium from a totally wild population. We've recovered our infectivity loci, we've recovered our resistance loci, and now we know the nodes, the network that connects them together. And we can also see, now that we have some confidence in this methodology, that this could very easily be applied to other host parasite systems, with the caveat that the more binary the outcome is, the smaller of a sample size you need. At this point, I had 250 whole Daphne genomes I sequenced. If you had a quantitative outcome, you're probably going to have to increase your sample size to deal with that noise. But in principle, it works. So let's talk about a rather interesting result that came from this and actually delayed the set of results for a very long time related to the E locus. So the E locus. For our particular population we're studying, this Lake Egelsee, we expected this to be sort of the major player because this is the locus which determines your ability to resist P20 infection. And we said that P20 is the dominant strain in this population. And so various studies that the group has performed using independent samples, independent methods to try to locate where the E locus is, they've all hit on chromosome 5, but when we try to localize it, these are all different markers that have come from different studies, and the actual location of the E locus has been really difficult to pinpoint, which has been a bit puzzling because the other resistance loci, the ABC locus, the F locus, we pretty consistently, through different studies, can get within a couple of kb every time. We know where they are. But for this one, we're looking at a window of maybe 7 megabases, where one study says it's on the left arm of chromosome 5, another study says it's on the right arm of chromosome 5. So what's going on here? So when I did a GWAS plot, this is that same set of 250 samples, and instead of doing genome to genome, now this is just simply the phenotype of whether the individuals could be infected by isolate P20. Because remember, I have clone lines for everything I sequenced. I can do these experiments after the sequencing. And so for this trait of being resistant or susceptible to P20, we see we've got clearly chromosome 5 is where the story is happening. This locus is on chromosome 5, but the signal is spread equally across both arms of the chromosome, which is, again, very unusual because we're dealing with a huge sexually reproducing population. We don't expect there to be these gigantic linkage blocks. So when I do a linkage disequilibrium analysis for chromosome 5, I find, well, actually, there is a huge linkage block. This is just a dot plot alignment of chromosome 5 against chromosome 5. That little dot there is the centromere. And we see that for most of the left arm, we have the signal that we would expect from a sexually reproducing, well-mixed population, being that the genomic correlation breaks up very quickly with distance. Meiosis and recombination is doing this all the time. But we have the right arm and a little bit of the left arm, which is basically functioning like one massive haplotype block. This is indicating right away that there is some sort of mechanism which is shutting down recombination over most of chromosome 5, which is interesting because even though we've been working in this system, working with a good quality Daphnia genome for many years now, this was completely flying under the radar. Trying to get a better sense of what might be happening to those, I started doing some dot plot alignments from whole genome assemblies, all from this one location. So multiple whole genome assemblies, all from one population. And when I take two different individuals, which have the same phenotype relative to this P20 isolate, meaning that they can both be infected by P20, when I do a dot plot alignment of chromosome 5, I get something which looks like this, meaning there's basically perfect synteny, sort of at the zoomed out view. If we thought, well, what are some other patterns we might see? If we had an inversion, it might look like something like this. If we had a duplication, we might have a pattern that looks like this. Or if we had something really unusual and we had each chromosome having some totally different sequence in the middle, it would look something like this. And in fact, when I take now and do a dot plot alignment from two individuals from the same population, which have different phenotypes, this is what I get. We see that these two chromosomes don't even look like they came from the same species. Over most of the chromosome, there is basically zero homology. And what's rather interesting is if I then do this dot plot alignment against many, many individuals from the same population, I find that when I do these sort of self-alignments, there are 3 different haplotypes, which are big enough haplotypes you'd be justified calling them different variant forms of the chromosome, all circulating in the same population. These are dot plot alignments. Also, I'm calling the structure the large structural polymorphism, the LSP. And if I do dot plot alignments, these are all different individuals, but ones which have different forms of the LSP. We see that we can always find multiple individuals which have the same version of LSP 1, 2, and 3. But then when we do pairwise comparisons, we find that there's basically this region where there is no synteny. So this is not the case of a simple inversion or some sort of structure where we have 2 variant forms. In this population, we have 3 variant forms where the sequence content is not shared at all. And if I look outside of this population, so far I've found at least 10 different forms of this, where we have 10 non-homologous regions spanning several megabases. Let's think about the gene content. So we know that recombination is basically shut down here. We know there's a big break in synteny. We know there's at least one phenotype that correlates with this, the ability to be infected by P20. But these regions can be between 2 and 5 megabases long. So is there possibly other phenotypic consequences? So to first off get a baseline measurement, I took 3 genomes. Each one has a different copy of this LSP haplotype. And I first looked at the left arm of chromosome 5. This is the arm of the chromosome which behaves normally. And we just do a Venn diagram. How many genes are in this control region, and how many of the genes are shared between the 3 individuals? And we see that about 90% of all the genes are shared between all 3 individuals. Now, if we look at the right arm at this large structural polymorphism and do the same analysis, we get the opposite pattern. We have, again, about 500 genes are located in this large structural polymorphism, and they're almost all private genes. Only 3% of the genes are shared across all 3 haplotypes. But otherwise, for example, LSP1 has 182 private genes that are only found in that haplotype. And so we discovered the structure through the phenotype of resistance to this particular Pasteur isolate. But clearly, there's going to be other phenotypic consequences, given that these are all genes which appear to be functional genes. So we've already filtered out things which appear to be pseudogenes. And then just sort of looking at sort of how well this maps onto phenotypes and whether we can figure out what the dominance pattern is. If we look at individuals here, which are containing 0 copies of LSP1, they're almost exclusively resistant to P20. Then if we find individuals which have one or more copies of the LSP1 haplotype, we see that almost all of our individuals which are susceptible are in this group. So this seems like a pretty clear case of simple dominance, where the LSP1 haplotype is dominant for the trait of susceptibility. We see some individuals that are sort of in the wrong categories there. These could be genotyping errors, or it could be that there's another sort of rare locus which is epistatically involved in this. But I think the overall pattern is quite clear. And then if we bring this back to patterns of seasonal selection now, we saw that there was this large change over the course of one season in terms of the phenotype of the ability to resist P20 infection. But now if we go back to that data set and we look at, again, that same year, and we just break it into 2 time points, individuals that were sampled in the spring before the epidemic, and individuals which were sampled in the fall after the epidemic. We see at the beginning of the season, all 3 of the haplotypes appear in almost perfectly equal frequencies. But after just a few short months, one of the haplotypes has basically disappeared from the population. This is the haplotype which was dominant for susceptibility. This is important that it's dominant because it can't hide in the form of heterozygotes. Basically, anytime it's present, the individual receives the phenotypic consequence of containing it. So we see that this co-evolutionary dynamic is happening really at the level of this ultrastructure, not at the level of a single SNP, not at the level of a simple indel, but really it's functioning on the chromosome itself. As an aside, even though these forms seem to be extraordinarily different, there's no evidence that they act as a barrier towards reproduction. So we find all possible combinations of the LSP in the wild. So it doesn't seem like you have any sort of lethality when you combine one of the forms of the other form. I think about covered everything there already. Let's skip to the more interesting parts. So I was really frustrated by this structural variant because it really slowed me down for about 2 years trying to understand why my GWAS consistently could not find this locus, which we knew had to be involved in this interaction. And at the end of the day, I found out it's because my pipeline was filtering this entire region out and just throwing away all of the data. So of course, I was never getting the signal at the end. And I started thinking about, well, is there a way this could be avoided in the future so I don't have to go through so much pain of sort of inferring there's a structural variant when after the fact, it was so obvious that that was the case. And so let's talk a little bit about reference genome bias and what it means from a very practical perspective in the terms of population genetics. So we have now, in sort of the modern era of genomics, the ability to produce multiple reference genomes. A lot of well-studied species have multiple well-curated reference genomes, which is great for when you're doing comparative genomics. But when you're doing population genomics, all of our pipelines and our workflows still force you to choose one to use as your reference. Because the way that our approach works is we have our reference genome, and then for any sample that we want to sequence, we shear the DNA into many, many pieces. If it's short-read Illumina sequencing, there may be 100, 150 base pairs long. If it's long-read sequencing, it's still little chunks. And we're going to align these all to our one reference genome. There is the possibility to do multi-genome reference, but it's not widespread. We then determine, okay, then the sequence of our sample that we're interested in green here, it must be this thing on the bottom based off of what we've inferred from our top genome. We run into the issue, though, however, if our sample has some sort of structural divergence, which is so great that we can't align reads to the genome. What happens at that point is this becomes a massive region of missing data, and our pipelines, well, if they're well designed, they throw this region away because there's nothing you can do with it. So this just becomes a big black box. But there could be really interesting biology happening in these regions, as we just saw with the study I was presenting on. So I started thinking about what signal could we use to try to understand that. And I found that genome coverage actually works really, really well. So we're looking here, this is from that study that I previously showed, where I had 250 Daphnia whole genomes I sequenced. And this is just a histogram of the coverage for each of those genomes. We had about 30x coverage, which is great, but we see there's quite a bit of variance around this peak. And it turns out this variance can actually be a really useful tool to quickly and cheaply find structural variance. What I mean by that is here now, we're looking at a plot of mean coverage. And this is mean coverage across all 250 individuals on the y-axis. And this is just position of chromosome 5 on the x-axis. We see there's a lot of noise here. There are some big dips in coverage, some peaks in coverage. But we see that the E-locus region appears as one of these dips in coverage. But of course, there can be lots of reasons why we don't have coverage. One of the classic cases is you've got a low complexity region. You've got a bunch of repeats, you've got a centromere, you've got a telomere. The regions are intrinsically difficult to align reads to. And so I was wondering, so when I have a dip in coverage here, is there a way that I can tell that the structural variant looks different than something like you see at the very far right there, which is the telomere? And it turns out actually there is. If we, instead of looking at the average coverage for all 250 samples, we look at the individual coverage and plot each one as a separate line. And we see something that emerges. This looks very similar to a plot that we saw earlier today, where we have at the E-locus location, we have 3 bands of coverage. What do those 3 bands correspond to? Well, the reference genome is a homozygote for the LSP1 haplotype. These individuals here, they have no coverage difficulties because they have the same haplotype as the reference genome. These individuals here, they have 1 copy of the same haplotype that the reference genome has, which means Okay, we're good. We're back on. So these individuals here are homozygotes for other haplotypes, and basically nothing they have aligns in this region. But if we look at the telomere region, we don't have this bandedness at all, because this is a complexity problem, and every individual maps equally bad there. We saw today that a really interesting way that we can identify this bandedness is to look at the position in the first principal component analysis. Really cool idea, I like that. The idea that I came up with, which sort of gets at the same thing, is we just look at the variance in coverage across all of the samples. If I overlay that, we see here we have this huge peak in coverage variance precisely at the location of this large structural polymorphism, but we don't have this peak at the centromere and at the telomere side. So this is really great. Now we have a signal that differentiates these 2 things. The other really nice thing is we always get coverage data for free anyways. This is something that just naturally is going to come to us through any population genetics workflow. So we don't have to design any special models. We don't have to use any special tools. The data's already there. Let's ground truth it against another locus. I'd mentioned before that we have another supergene, which is known in the Daphnia magna system, the ABC locus. If I apply this same statistic to the chromosome where that locus is located at, we see it pulls it out immediately. That peak right there that you see is the location of the ABC locus. So it works. That's great. Do we have other spots in the Daphnia genome where we might have supergenes? It looks like it right here. I have no idea what this is. We've been looking at this large structural variant for many, many years, but it's just never come to our attention before. If I just run this simple test across all of the chromosomes for all of the Daphnia genome, and I put just an arbitrary red line, has no intrinsic meaning, just a visual marker, you see that we have pretty compelling evidence that we have these huge structural variants on basically every chromosome in the genome, oftentimes multiple cases. If I sort of sum these all up together, it looks like about 1% or so of the Daphnia magna genome is comprised of these types of large structural variants where you have such great homology that you can't map, which means you cannot call variants inside of those regions. They're functionally invisible to any population genetics you would do. So from this we can say reference genome bias is a very real concern. I spent about 2 years spinning in circles precisely because of this issue. And I was only able to overcome it because from this population we had a variety of whole genomes. So I could do a multi-reference alignment, but that's not a luxury everybody has. We can see that this is, for this species, particularly relevant. This is not just some rare novelty that I've discovered. We see that the genomic variation is completely invisible to our pipelines when we're using a standard variant calling approach. And this doesn't require any sort of special experimental design. You can do this type of analysis after the fact with a data you've gotten from GenBake. Anytime you have coverage data and a data set, you can repeat this analysis, and so it's cheap and it's easy. So how does this relate to biodiversity and cogenomics and other projects more broadly if you're not interested in Daphnia or parasites or coevolution? I'd say a couple of ways. One, host, parasite, symbiont genomes, they're often sequenced together along with your target. Usually our focus is on getting rid of these and minimizing them, but typically our pipelines throw all this information away. That can be useful if you have a very narrow focus, but there can also be really useful information that we're discarding there. We see that structural variants, as I said before, they're probably a lot more common than we currently consider, and we very often overlook them, but they can be relatively easy to find. And that these coverage inconsistencies, which are treated as sort of technical artifacts or nuisances, can actually be a really powerful tool to quickly and cheaply understand something more about genomes that we already have. So I have no idea how I'm doing on time, but I'm sure no one's giving me any dirty looks, so I think I'm doing okay. I have to huge acknowledgement to Professor Dieter Ebert. So all of the work I presented here is work that I did while I was a postdoc in his laboratory. So he's been running this Daphnia lab for much longer than I've been involved in the system. So excellent mentor, excellent friend over many years, and I still have continued projects going on with him in the future. The work was supported by a Marie Curie Fellowship to me, and then continued afterwards with a Swiss National Science Foundation grant from Dieter. And then finally, as mentioned before, so I am now a freelance bioinformatic consultant. I work with industry, but also research laboratories. So if you need a bioinformatician or you just want to talk about crazy ideas you have, you can always feel free to get a hold of me anytime.
Thanks, Eric. Really nice talk. Questions for Eric? It seems to me that we are now
Everybody wants to go home.
No home. We have beers. No, you are not allowed to go home. So, some questions. It's clear to me that now we are in the age of structural variation. So, scary.
Hi. Well, nice talk. Just a curiosity. I mean, in one season, because the Daphnias goes, the season is in spring, basically. Mm-hmm. It goes in spring. How many generations do they have in one? Just to see that change in these gene frequencies. Just to figure out. How many generations or what's the generation rate to see that quick changes?
So the generation rate is temperature dependent, and the season goes from about April until about September. So you could say roughly 10 would be a reasonable estimate for each year. 10 to 20 maybe. They can one generation can be as short as say 3 to 5 days. That's only in the peak of summer when the water temperature is really warm. So, but it's a very short time in generation time and in real time as well.
Hi, very interesting talk. So you have a two-party system. What happens when you add more elements to this system? Because very often animals are not infected by a single parasite.
Exactly. So it depends a little bit on whether or not there is overlap in the sort of the parasite and the mechanisms. So one of the things that I didn't talk about is that part of what we believe maintains diversity, why you maintain susceptibility in this population, is because there are other Pasteuria sort of clades, which the susceptible genotypes are resistant to. So basically, when the population fixes resistance, suddenly now they also fix susceptibility to a different clade. That's within one parasite, Pasteuria, though.
Very interesting. I was wondering about this standard deviation in depths. Actually, this is something I've also used, but only for filtering and then threw it away, so not looking at it. But isn't it quite? Do you have a good way to defining a threshold for the significance? Because as you showed, there is a lot of variation, and it will depend on the allele frequency of the different variants, like how strong the signal is, right?
No, I don't, but there's a pretty easy way to get that threshold, sort of in which that you sort of, you run your analysis, you find a cutoff value, and then you just start looking at your peaks, and visually the sort of the bandedness is readily apparent, and whenever the bands sort of stop appearing, then you have confidence you found your threshold. I tried doing a bunch of statistical methods to sort of quantify the bandedness of the signal there using all sorts of Bayesian models, but I didn't have a lot of progress there. I found that visually doing a spot check got me there really quickly.
And I mean, one issue is that, of course, you rely on at least one copy being present in a reference genome. Otherwise, you wouldn't align it in the first place, right? So did you also look at the reads which didn't map to the reference?
So there is a big hindrance there in that if you have a reference genome which has a haplotype which nothing in your population has, the variant remains invisible, which is why your suggested approach of going through PCA in that case actually I think is the superior approach, because this is not great identifying rare structural variants because it's not going to move the variance very much, and if nothing aligns, nothing aligns. But I think in those cases, I think using the first principle component would solve that issue.
One last question. Okay, if that's not the case, then thanks again.
Thank you.
So just very quickly before we leave, we wanted to thank again everyone for coming, for being here today. Of course, thank you to all our funders, especially Conexión Genoma, but also the Company of Biologists, Novogene, and the Sociedad de Amigos del Museo. Thank you very much to our wonderful speakers. It's been a pleasure to have you here and to learn from all of you. And thank you all for coming. And now, as Iker mentioned, you're not allowed to go home yet. So please join us in the garden. We have some time for mingling and talking over some beers and drinks. So thank you, everyone.